Good morning, everybody. Welcome to this second edition of the Navigated TMS-EEG workshop that we have organized also 2 years ago. This is our second time. Welcome to all of you who came in person to attend this meeting, but also welcome to online attendees. I know there are many online attendees, so welcome. I hope you can hear me well. And we did our best to allow you to follow effectively all the activities we will perform today in this room. There will be theoretical, let's say, teaching classes, but also practical sessions with real TMS-EEG hands-on experiments. Organizing a workshop like this is always very challenging. And of course, I'm not alone in organizing this. So I want to thank Professor Mario Rosanova and Dr. Gabriel Hassan, who composed together with me sort of organizing committee. But we received the help from all our colleagues, and you will have the opportunity to meet and know them better during the workshop, because they will help with the hands-on session, with the teaching sessions, and I want to thank all of them for their help. And I also want to thank Brain Products and Brain Products Academy. You all have registered on the website, so you already know that they have been part of the organizing team. And I want to thank also nTMS company, who supported the workshop by providing some equipment and support in general. So thanks everybody. I hope you will enjoy it. And I would like to make the sessions, if you also agree, quite open. So if you have questions, just please raise your hand, interrupt, and ask the question you would like to ask. Thanks a lot. So the first speaker will be Henri Hannula by nTMS, and I want to thank you very much for being here, and thanks a lot.
Yeah, thanks a lot, Silvia, and thank you for the whole team. As always, it's interesting to see new methods coming live and then to be developed. But like this workshop is all about seeing how it is done, and seeing is always believing. That when you have non-invasive method that you can use to activate the brain, and at the same time you are able to measure the responses from the cortex, it's fascinating and opening new doors in the whole neuroscience, and then later on to the clinical applications. I'm extremely happy to be in this workshop now second time to give a talk about TMS, and particularly there the navigated TMS. So the history of nTMS has been actually quite interesting. One and only meaning why we exist was the idea of Professor Risto Ilmoniemi to make first time ever the transcranial magnetic stimulation visible. By the idea that if we can make the transcranial magnetic stimulation visible, it would make it also accurate and reproducible. And there is two terms, accuracy and reproducibility, meaning precision, what we come later on, which are interesting for you maybe also for the next days when you are using the device and seeing what's happening at the cortex. So how you can target the pulse. Let's get started. So the whole content here will be introducing first the transcranial magnetic stimulation in neurosurgery. And why is that? Because maybe not all of you have seen all the data that he's published and how neurosurgeon has used this non-invasive method to map the brain before the surgery, the most critical application you can imagine in the world. So with that example, you get an idea how precise and accurate you can stimulate with TMS. But there is a lot of parameters you need to take into account. But let's look first a couple of videos about how this is used for motor and language, and then how it is used in neurosurgery. Once we have seen that, we'll go for the theory of TMS. So how TMS stimulates the brain, what is activated and what are the mechanisms to understand again what is the accuracy of TMS. when you use the targeting based on the stimulating electric field. And then at the end, the role of navigated TMS to collect the data, and that will give you a nice introduction for the upcoming talks about the TMS-EEG applications and the study setups that you can plan. And like Silvia mentioned, feel free to stop me anytime and make questions. So first video, you will see, this is very quick, by the way, I don't bore you with the long videos. There is motor and language mapping principle given to you in one video. So what you see here is actually a tumor case. You can maybe recognize on the right side image a huge tumor located close to the motor cortex. The question is always, before the surgery, where are the functions? There is anatomy textbooks from the healthy brain. All of us are individual, but particularly when there is a tumor, this creates plasticity. The functions are changing. So how to map those functions before the surgery? So we apply the stimulus with the foot pedal and can measure the responses from the muscles. The amplitudes are given, and those are used to color code each and every stimulation spot. The brighter the color, the bigger the amplitude. Then for the language processing, when you hit the right spot, you can even disturb the language with the train of pulses. So you saw just one single pulse to be used actually for motor mapping, and then a train of stimulations that was used for language mapping. Then you may ask how this data is useful, how it is used in the surgery. Let's look another short video how these results are used. And this is from Technical University Munich at their neurosurgery team. Right here. So when the mapping is done, they export the data set to the surgical planning software. And again, another tumor case here in critical area, particularly for language. These white dots, what you see here, are the dots for positive motor locations, different muscles. And there was quickly presented the positive language spots. Now these are brought to the surgical planning software. The green ones are for motor mapping. And then they look the structural tractography from those seed sites. Where are the connections? The same for the purple colored language positive maps. Where are the connections starting from those locations? So the navigated TMS is used as a seed site. And if you now look as a neurosurgeon, the orange tumor that you should resect, you can turn it exactly, you're gonna plan it in the surgery. And now they confirmed only in 10 minutes the language area, it was as they planned. And this already is end of surgery where they confirm the borders of the resection, and this is post-operative image, and there was no functional worsening. So this gives you an idea how to bring motor and language data with navigated TMS to neurosurgery. How does it work? First, you upload the MRI image. Of course, MRI is structure, it's not function. Then you align that with the physical head, as you will see now in these demonstrations, and test by yourself. You stimulate by navigating with the stimulating electric field where you want to touch the brain and how much. And then you can see the motor functions or language functions, and you export them to the surgical planning softwares, like offered by BrainLab Medtronic Stryker in the operation room. And you may think that, is it reliable? And this is from one example and what value this can bring to the neurosurgeon. If you look these patient cases from A to F, they look very, let's say, similar and challenging. All tumors in the eloquent area of the cortex. And of course, the question of you as a neurosurgeon would be, can I operate? Where can I operate and how much? Where are the functions? It's a difficult question, but if you add the navigated TMS data and these positive mappings here with the negative mappings, you can see that from A to E, you could actually go in safely and operate, but in the case F, you can see positive colored spots top of the planned tumor resection area. But even that case is not like end of the story because of the plasticity. You may resect some part of the tumor and then map again after the surgery, some months, weeks, even half a year if you have time, and then you can see that the functions have been moved away and you can operate again. But let's look more detailedly one of these cases, like the case B. So you can see that there is close to midline the leg area, and then other side of the tumor there is a hand area. Cloud of stimulations colored from white to red. But even more important information is at the top of the glioma, the resection area, which is all gray. Those gray dots are negative responses from TMS. So actually you as a neurosurgeon are very thankful not only from the positive map, but for the negative map, where you can see that there is no function. I can go to the surgery, confirm the situation, and go and resect. Because of that information, this young patient, for example, imagine 20 years old patient only, no neurological deficit. And in the left image, you can see that the glioma is sitting right middle of the motor knob. And based on the somatotomy textbook, you would think this is a hand area. But you are very much thinking if you would go along or not with the surgery because there is no deficit yet, so should you wait? The longer you wait, the worse it is for the patient. The earlier you can resect, the more cross-total resection you can do, the better for the patient. So this data helps helped in this case that neurosurgery team to decide, instead of waiting, immediate surgery, doing that with the tractography guidance, and see the cross-total resection in the middle, only transient deficit. Transient deficit means that they were really on the border of the functions, but the patient recovered afterwards. And that was from Charité Berlin by Dr. Thomas Picht. So how about the language mapping? Similar to motor, you do the registrations, but then you show the images to the patient and they name objects. Once they name the objects perfectly without stimulation, you add only those images to the procedure they named the same way 2 to 3 times. And then you disturb the processing by stimulating with trains of stimulation like 5 Hz, 5 pulses, 7 Hz, 7 pulses. And then after that, you do the analysis between the baseline without stimulation and with the stimulation and look the differences. And then report those differences. Then you may ask, does it have an impact in neurosurgery? Because you know that there is also methods used in the surgery where you use direct current stimulation to stimulate during the surgery. If you add this data before the surgery, it has been shown that even 35 to 40% more cross-total resections are completed, so the whole tumor has been able to be removed if you use the navigated TMS data beforehand. And then you may even ask, does that have an impact? And if you look the progression-free survival months, it's also increased like 46%. So it has a huge meaning for the patient and the surgical outcome that you know the functions beforehand, even you use direct cortical mapping during the surgery. It's not only these data and examples I showed you, there is already thousands of patients in these, more than, I think, 170 articles already these days, so there is some outdated information here, but it's totally shown that in different neurosurgical indications, this helps with the resection, and it actually helps to us understand the accuracy and precision of this method. So now we have seen how it has been used in neurosurgery, but still the question is how it is possible to target the TMS and what is the accuracy of TMS. You stimulating now here soon a subject brain, what is the resolution that you are expecting to get? So first of all, what is TMS? It's interesting that the term is transcranial magnetic stimulation. Since 1985 by Anthony Parker, when it was published, the method was clearly indicated as magnetic stimulation. But what happens there actually is that we create a huge current pulse through the coil, and that induces a magnetic field. But it's not the magnetic field that stimulates. it's the change, the rapid change, like 30,000 Tesla per second. That change will create an electric field, and that electric field is stimulating. That's already mentioned in these 1985 papers. People just forgot it, that the E field is the key where we stimulate. So when you see where the stimulating electric field is hitting the brain, you know where you are stimulating and how much and which orientation. And we come later to that importance of orientation of the electric field as well. So how we stimulate and what is actually the E field there? If you look the induced electric field from the coil, we use the tip of the cone, let's say in here. The more intensity you put to the TMS, the deeper you push the cone. If you want to blast the brain with the full intensity, you can stimulate large area, but then you lose the resolution. So how we can show you then, or what would be the right way to think that what do we stimulate? If you look now, when you operate with the system, the stimulating electric field, you understand it's the top of the, or the tip of the E field, the maximum, which is indicated in red over the cortex. But how does it affect in the brain? Now, if we take a very close look to the tip of this electric field here, and you can see that there is a primary motor cortex and sensory cortex right next to each other. And we put the electric field perpendicular to the gyrus. And we take even more microscopic view. You can see neurons there, and you can see axons. What do we activate and how? If we take even more closer look, for one individual axon, with the orientation of the electric field, what happens there is that actually we cause a local depolarization at the bend. And why is that? In that part of the axon where you have actually this, let me see here, pointer. This part of the axon, which is aligned with the E field, you get different voltage than this part of the axon, which is actually other direction in the E field. If there is a voltage difference in the axon, which is like conductive cable, there is no voltage difference that can exist in nature. They need to equalize. And that happens at the bend, and there is polarization at the bend of the axon, and then the signal is firing. One single axon doesn't make the movement or some kind of reaction, but when you have group of these actions and dominating orientation and bundle, and you go with the maximum electric field, like my fingertip size, and you probe the brain with different orientation, you get actually the accuracy and precision you would wish to study the brain in different areas. So strongest activation is always at the maximum of the electric field. And there is complex cell shapes where this direction is important. The good thing is you don't need to believe me. You can test it in this workshop. You can take the coil, stimulate the individual healthy brain in this case, and you can see how the response is varying. When you put everything correctly, everything, the E field is nicely there in the perpendicular to the motor knob, the tilting of the coil is perfect, you maximize the electric field, you get huge response, like 1.48 millivolt. This is not even needed, that kind of amplitude, because it would be already a concrete movement of the muscle. We don't need to go that high, but let's say in this optimum case, you could get even this type of huge response. What if you are not correct? That's the one error that you may do. You place the coil perfectly where you want to, but you tilt the coil. In the top left corner, you have tilted the coil left. You have orientation the same, the E field location the same, the tilting is not correctly, so the E field is decreasing, and you have flat line, there is no response in the EMG. If we take the top right corner, you have placed the coil again, the same spot, same orientation, optimally that you got the huge response, but you tilt the coil forward. The blue bar is showing you where do you tilt wrongly. And then decrease the intensity, and again a flat line, no response. So the stimulation actually depends on the tilting of the coil. And if the tilting is changed, there is no stimulation response like motor evoked potential, even in the exact same spot. And this can be demonstrated nicely. What about the orientation? We just saw the basic principle of the physics, how TMS stimulates. If we take the optimum spot with the optimum tilting, everything, but we orient the electric field not perpendicular to the gyrus but differently, again, no responses if you turn to the left, like a top left corner, or if you turn to the right, like a top right corner. So the stimulation depends on the rotation of the coil. And if the rotation is changed, there is no response. If you control everything that, and you get the nice spot with the nice orientations and tilting, you can reproduce everything what you see here, like a huge response. You have the method itself in control. You may think, how is this possible? The modeling itself is quite easy. That's the physics that we have published already since 2003. But actually the challenging part is that how do you adapt that to the individual head shape and size? We need to take into account all these parameters: the stimulation intensity, the stimulator parameters, the coil shapes, the location of the coil, orientation, tilting, distance to the cortex, and of course the head shapes and size. If you control all of those, and the stimulator parameters include many things from the coil and the electronics, then you can get this kind of resolution. Again, this is a heat map from motor mapping. The brighter the color, the higher the amplitude. The darker the color, the lower the amplitude, and gray one, no responses. So you can really use like your fingertip to probe the cortex and map the motor functions there with the EMG. How does it feel like? You may feel like a fisherman with a navigated TMS, but if you are an unexperienced fisherman, you trust your eyes. If that would be a straight line, this is what your eyes will be telling when you are fishing. The straight line to the fish, and you would be hitting where the red arrow is pointing, you would never ever get any fish. You would miss the fish because the light is bending in the water. If you are experienced fisherman, you would know that the light is bending. I cannot trust my eyes. I always hit a little bit frontal, and then you catch the fish. Why I tell this is that it's not the same, but very similar with the TMS. It depends how you place the coil, how the individual head is below the coil, and this is shown in this animation. Like I mentioned to you, the whole idea of the company nexTIM by Professor Ilmoniemi was to make this visible. So what is the difference of navigating the TMS coil as an instrument and draw a line to the MRI? Remember the fisherman again. We'll take the primary motor cortex and primary sensory cortex. They are nicely right next to each other. And we place the coil one spot first, and then look where the line projection from the coil is hitting the MRI. And then we give a big pulse by seeing what happens in this part of the cortex. And there is the M1, the primary motor cortex, and S1, the primary somatosensory cortex. In first location, the operator, when using this kind of line navigation, would think, I'm getting huge response from the primary motor cortex because my line is pointing there, exactly like the fisherman. But the E field is hitting actually the primary sensory cortex, which explains that there is a flat line in the EMG, even you have 99 volts per meter. So there is no response because you're not hitting with the electric field the motor cortex. Then we put the same dose, but we control with the coil so that the electric field is hitting actually the primary motor cortex. Again, the line projection would be now in primary sensory cortex, and you would be shocked because there is a huge response if you would rely on this fisherman example. So the E field is telling you where you stimulate, and there is over 1 millivolt response. And this you can test when you use the device during the course. By remembering that, there was a question, what is the accuracy of TMS? Before going to accuracy, it's very important to separate two terms that are mixed nicely in literature. People talk about precision and accuracy without understanding the meaning of them. Accuracy itself means how accurate you can hit the spot that you wanted. In this case, the center of this graphics. The precision means repeatability, how you can hit the same spot, how precisely every time you want to stimulate it. If you think the whole literature of TMS without navigation, you would be the second right image, where you don't have precision, you go somewhere roughly, you stimulate, but you don't know where, so you don't hit the spot exactly as you want it, and you cannot reproduce that. So it's always a different location because your coil is moving, you're tilting, you are not controlling the parameters. If you think again the fisherman example and the line projection from the coil, you could have good precision, meaning you could reproduce inside of the session very well the spots, but you don't hit the spot that you wanted to stimulate, not the center. With the E-field guided navigation, you can get both precision and accuracy. So two terms important to note every time when you read literature or do your studies. But still the question is, what is this precision and accuracy? What can it be? There is a huge literature, and that has been concluded actually a book. There is a second edition published soon, but actually the conclusion of all these neurosurgery literature is that the spatial accuracy of those E-field navigated TMS, what we have developed, is better than 5 millimeter, and the repeatability, the precision, the targeting is 1 to 2 millimeters. And this is sufficient for pre-surgical localization of eloquent cortex, which is approved by CE and FDA. So that gives you an idea, and again, if you want to imagine your fingertip probing the brain, that gives you a good idea. Now we have had all these theory and clinical examples of TMS, but what does it mean for EEG studies? First of all, this paper is good to use as a conclusion that when you use this type of navigation by controlling the pulse, you can ensure the reproducibility of your studies. You take the technical variation of the TMS method away. And then with this real-time EEG visualization, you can maximize the output for the EEG signal without changing the output of the stimulator. Imagine you adjust something from the stimulator, but depending how you handle the coil, it affects how much you stimulate at the cortex. If you don't have that in control, you may have unoptimized dosing. And you can let not the EMG, but the EEG guide your response. So the luxury of this workshop is that we don't need to measure the periphery. We can measure directly from the cortex and see how that responds. We are not limited to motor cortex. We are not limited to language cortex. We can measure the whole cortex. And the neuronavigation itself, key for reproducible and reliable responses. How can it be seen in the real life? And this is very nice visualization and concrete example from it. If you think now this top image where the blue arrow was used to optimize first the response, you can see the response how it is in the right side. If you take again the same spot with the same E field, but you turn it at 60 degrees, the response is much different and amplitude is decreased vastly. So controlling the orientation, you can change the amplitude what you see from the cortex. If you can control this, and if we go now to the lower image, 1 week apart, and you optimize, you stimulate the same way like in the blue arrow, you can see the blue and red response 1 week apart, same subject, being totally reproducible, highlighting the importance to control the TMS variables with EEG. This is another example from stimulation angle. The previous was with 60 degrees, but if you look now the left image, where it has been used like one optimum condition, but then the other one turned on the left 30 degrees, you can see the amplitudes in the left and right side, which are like day and night. So don't be happy with one location with your EEG when you move the orientation even 30 degrees. And believe me, the 30 degrees sounds to you that it's very huge movement. It's not. When you start operating with the coil, you can do 30 degrees movement like with the small change of the handle. You almost cannot see it if you don't navigate it. And you can see in the amplitude how huge difference there is. And of course, we will hear later on about the auditory masking a lot, but there was an example that if you look the left side image and the right side image, in the left side image, you have the lower intensity without noise masking. The signal is looking totally disturbed compared to the right side image, where you have even increased the intensity, but used the color-coded noise masking, getting those real responses visible that are not masked by the audio response and the second, the left side image. So auditory masking, we will learn a lot how important it is, but this is just to highlight you that it's not all about the TMS variabilities, it's all about the noise masking as well to get reliable responses from the cortex. So where does this can lead to? And this is what we learn in this workshop with the hands-on. So you can actually get a tool which you can stimulate and probe the brain. And it has been shown already thanks to the team of Milan with coma patients, how you can measure the consciousness reliable and repeatable based on the index they have developed for that. And that index is reproducible after the teams have been trained and have the right tools to do it. You can actually measure the consciousness reliably from center to center and patient to patient. And this offers us now a new tool to predict actually the functional outcomes and guide the intervention. And this is not only limiting us to the consciousness. Imagine that there was a recent publication actually last week from one of our neurosurgery sites for pediatric patient for post-operative rehabilitation. So after the surgery, after the complications there, they had actually a bleeding and severe paresis. And they started to stimulate that young patient 1 hertz, other hemisphere to inhibit the healthy side to let the stroke side to recover. And they made the physiotherapy top of that. And they got actually the patient much better. So the kids started to move again with the hand and the legs. But then the publication says still that it would be highly interesting to see if we could show what the effect of TMS was. And it was a clear indication for EEG. Imagine if they would have had the EEG and they could have measured the amplitudes of the both hemispheres before and after, they would see what kind of effect TMS brings to the cortex. Now they could see the effect in the periphery. But how much of that was TMS? How much was that to the physiotherapy? It depends it depends in the case by case and is under the discussion. It's not only this single case. There is of course series of adult patients that are published even in randomized controlled study, and there is actually a clinical application coming now about this for rehabilitation. But this same way of measuring the excitability of the cortex could be used also for the stroke patients, as it has been done successfully to predict who will recover and how well. Do you have the right parameters? Do you have right location? Same for depression, pain. Actually, all central nervous system indications would be really interested to see what kind of excitability changes there is when using different stimulation targets and different parameters to guide the therapy. This was now as an introduction. There is plenty of more beautiful talks coming about the method itself. We have also collected some of the highlights on our webpage that you can visit later on and look the talks from Silvia, Marcello, Pantelis, Liomis, and Laura Marchetti about these important methodological papers and outcomes, how it is used in the science and for the patients. And I thank you for your attention and happy to receive any questions you may have.
Questions from the audience?
I mean, the beauty of the workshop is, of course, that you don't need to understand everything right now here, but when you go to the device and you use it, that's when you start to get it. And this is just the basics for you to understand what you see then when you are using.
Sorry, just want to ask you one thing. You talked at the beginning of your presentation about the surgical applications for TMS, for pre-surgical mapping of functions. But also recently, the EEG part has been also integrated into this application, which was more TMS only at first. Can you comment a little bit about this, about what EEG is adding to this?
Thank you for the great question, and it's a good example that this method has been seen to be very complicated, very unreliable. And now, thanks to the Milan team and other our key users, they have brought it to the level that you can use it as a clinician for your patients. And there is actually quite a community now who is using this for neurosurgery research with one very important reason. You could maybe do this for cortical mapping. So not only looking at what happens from the cortex with connected to periphery, but you could see for the tumor patients how the cortex is working as a network. And that could create more valuable information for the surgery. Particularly important, of course, in epilepsy as well, and epilepsy surgery. But this huge interest for cortical mapping has been increasing now. And I think it was last October we had a big symposium and the audience voted all of the neurosurgery presentations from abstracts the most interesting poster, and that was actually the TMS-EEG poster and that mapping. So the neurosurgery community is very much looking forward to this method.
She was quicker than you, François.
Thanks. Hi, thank you. Just a curiosity, so instead of an area, could you target like a network? So instead of just having the MRI, could it be possible to compute like connectivity map and use it like in the neuronavigation system? And instead of targeting like just one area, you target like a network? Thank you.
So you meaning using connectomes, or? Yes, absolutely. That's a very interesting part of the science that is now developing towards the clinical studies as well, that would use the connectomics and see the hubs that are cortical and connected to the deeper structures. And then you could go with the E-field there in those hubs and stimulate and maybe measure then with the TMS-EEG the responses and the connections between different cortical areas. So indeed, that is very interesting neuroscience field currently, and I think we will see more and more about that kind of measurements.
Thank you. And then another curiosity. So of course, the motor and the language function, with the neuronavigation addition, was there any like advance in like mapping other areas that they were more difficult to reach with the TMS? Because with the motor and the language, you have the output of the behavior, so it's something easier to monitor. So does the neuronavigation make it more possible to, I don't know, stimulate maybe an occipital cortex or other areas of the brain? Thank you.
Yeah, very good question. And again, like you were mentioning, limiting us to the motor cortex, you could put even without the navigation, you can go with the coil in the area of motor somewhere, stimulate and use a response. Of course, the problem there is that you don't know if you stimulated sensory cortex, but just higher intensity, and you happen to hit also motor cortex. So you don't know where the response is going, but you can activate the motor cortex because of the direct feedback. But like you say, the real value as well could come in the occipital cortex, where you have a visual areas. And there has been studies that have been able to cause these phosphines reproducibly by targeting the E field in the right areas. And now there is some publications already from neurosurgeons using those sites as a seed sites to make the visual system visible. That's one example, but actually all cortical functions could be now mapped even indirectly. Now with the EEG, you can measure the connectomics and the connections, and you can test your theory for that particular patient group or indication.
Thank you.
Thank you for that talk. I'm not just quite sure that I get the first part of it, like, because it was like, okay, if I don't get like a function with the TMS, like either like speech or movement, then I can remove part of the brain with the neurosurgery. So it was like, is it like, okay, if I don't get function, I can just remove more to make sure that the tumor is removed? Or because it seems like we assume there's no function if there's no like behavioral output. But you mentioned TMS-EEG, so I guess that now we have more like, as you said, seed or like matrix that you would say, okay, no, if I don't see this P60 or N100 or something like in TMS-EEG, I won't remove that part. So just maybe, yeah, if you could comment on that.
Yeah, thank you. Good question. And we have a really good, actually, participant in the auditorium as well as a neurosurgeon that can help to answer, and you can discuss later on about the details. But yes, you map motor and language, and those are like very important for the patients, of course, and you can make them visible here. But what they are also doing in the operation room is more advanced mappings in some locations where you ask, the patient to perform a task where they move and talk and interact them much more advanced than just basically object naming or motor. However, not every patient is able to collaborate during awake surgery, so that is limited, or if you have pediatric patient and so on. So there is tools as a neurosurgery teams who are experienced for awake surgery to map like through functional combinations or complicated tasks during the surgery if the patients are aligning with that. And that leads, of course, even more aggressive resections. Like Professor Hugues Dufault in Montpellier and Sam Ng is here in the auditorium. I mean, it would be amazing if you can talk and learn what they operate and how. So it's like, again, the somatotopy textbook, you can forget it. I mean, you can resect tumors like size of the hand in the left area where the language should be, or the classical Broca area. But it's through the plasticity changed, and it's very much individual treatment. The most important thing here is that this motor and language, 40 to 50% of all the patients that have a tumor, that map has changed. It's not there where you are assuming that to be. And that plasticity can be done visible. So the EEG now, adding to that, could tell not only of this, but the network function that where the whole brain is working with this, cognitive task and so on and so forth. So there could be a much more advanced information. There is also, we are lucky because cortical mapping is also developed in the operation room recently with some groups. And that offers us also more information to compare. And then these very, very advanced awake surgeries will tell us also about the plasticity, which you can use as a scientist as well as a reference when you do your studies.
I'd like to introduce you Sara Pizzamiglio from Brain Products. She will talk about simultaneous TMS in EEG. How does hardware influence the quality of the recordings? Thank you, Sara.
Thank you, Silvia. Hi. Hi, everybody, also from my side and from the whole Brain Products and Brain Products Academy team. And hello also to those online. Thank you for joining us. So, disclaimer. This is going to be a little bit a technical presentation. So, why? Because when we do science, when we do research, we like to have very nice data. We like to have high quality data. But in order to get high quality data, sometimes, if not all the time, we also need to know our hardware. So we need to know with what we are working with and what are the technical specifications of our hardware and how those may influence also the quality of our data. So sometimes we give these things for granted, so we don't look into that. But coming from an engineering background and working for Brain Products as a company, I know how these then can influence quite a lot the quality of EEG data, especially in challenging environments like in simultaneous TMS-EEG. Okay, so this is going to be a little bit technical. I'm gonna try to speak slowly, and then you will have the time to ask questions, of course, if something is not clear. All right. Before diving into that, let's know each other a little bit. So my name is Sara. I'm originally a biomedical engineer, and then I went into more the neuroscience with my PhD, work with EEG and TMS as well. And now I work for Brain Products in the sales team, and I try to promote solutions for EEG as well as for different type of applications like TMS-EEG, conferences, workshops, so trying to teach and try to promote our solutions to researchers all over the world. So this is me. That's me actually at the Brain Stimulation Conference in Japan this year. How about you, both you in person and online? So raising hands. Who has worked with EEG only? Raising hands. High hands. Raising hand. And people online, there's a button that you can press. Only 2 persons? Only EEG? 1, 2, 3. Okay, 7. Wow, okay. And who has worked with TMS only? A little bit more. Nice, okay. Who has worked with both? Interesting. Okay, so that's the right place for you. So hopefully 2 persons online. Okay. So hopefully my talk will give you a little bit more insights on the EEG part. So what, especially what an EEG amplifier looks like and what's inside an EEG amplifier and how this influences a recording with TMS. So let me start by showing you what was originally a EEG amplifier. So this is the very first amplifier used by Hans Berger back in the 1920s. That was the very first experimental type of EEG that was done. As you can see, is really big and it's full of mechanical parts, and so completely different from what we are using nowadays. But most importantly, the participants at that time, they were patients that had undergone trepanation. So EEG was recorded intracranially, basically. So that was quite invasive. And I like to always think about EEG that was recorded analogically. I like to think about it like the machines that you use to detect earthquakes. So it's like you were really depicting the EEG signals on a paper, and then you were using a ruler to measure it. So that was quite exhausting. But thanks to technological development, we are now able to record EEG with much smaller devices. These are 2 examples from the devices that we have, but for sure other EEG manufacturers that you have worked with, you know that all the EEG amplifiers, they're quite small nowadays. And Thanks to technological development, we can now record EEG non-invasively, fortunately, so we don't have to trepanate patients anymore. The recording is digital, it's not more on paper, it's not more analogical, everything is on a computer and can be then saved, analyzed. Usually preparing an EEG setup and doing an EEG experiment is quite comfortable, usually, depending on the experiment. The technical specifications of the amplifiers nowadays are also pretty high. So we have moved quite a lot from this to to here, okay? Especially today, considering the TMS-EEG combination together. So this is the history of how we started and where we are with EEG technology. But let's have a look at what's inside an EEG amplifier, okay? So an EEG amplifier is, by definition, a device that is capable of recording continuous signal in time from the brain. Inside the EEG amplifier, there is an analog to digital converter that transforms the data from continuous in time into discrete in time. And the discrete in time signal is then saved on a computer. So the amplifier takes the data, transforms them from continuous to discrete in time, amplifies the signal, of course, also the name amplifier comes from that. So this is more or less what it does. It looks simple. So we go from a time, from a signal that is continuous in the time domain to a signal that is discrete in the time domain. This looks easy, okay, this looks quite straightforward. However, inside an EEG amplifier, there are many more parts. There are many more electronic parts, electronic components that may influence the quality of the EEG that you're recording, depending on your application. There are some applications that are less prone to be sensitive to particular artifacts. For example, recording EEG in a lab when you have simply a visual stimuli at the computer and the participant is just sitting and relaxing, that's pretty straightforward. But when you do it in combination with TMS, for example, there are some things that we need to consider. So there are some things that we need to look out, okay? And so let me start also by saying that this part here, before the ADC, is all hardware, so they are all physical pieces, okay? Whereas everything that comes after the ADC is all done by the recording software. In our case, by Brain Vision Recorder, with other manufacturers by their recording software, okay? So there are things that are done on the hardware side, so in the amplifier, other things that can be done by the software itself. Now, I don't want to spend too much time on this slide because we are going to go into each and every most important feature of an EEG amplifier, of how we record EEG, one by one, okay? So let's start by talking about the differential recording principle. So you may have heard manufacturers saying that the EEG amplifier follows a differential recording principle. What does that mean? So the EEG amplifier, by definition, takes two signals in input. So what you see as a signal on your recording screen usually is the difference between one signal that is recorded from a specific point on the brain and then another reference signal. And by default, usually all the amplifiers take the ground as the first default reference signal. However, the ground, since it's connected to the ground, contains a lot of artifact, of common mode noise artifact. So in order to prevent these artifacts, this noise, to spread all over the other channels, we add one more reference. It's called what we call the reference channel. So this is why adding the reference channel, basically what we get at the end, what we see when we are recording online, or what we see when we are analyzing the data offline, is the difference between one signal, one channel, and the reference channel, okay? Simply because of a simple difference here, okay? So this is very easy. This is a principle that you can keep in mind. So when you hear about differential recording principle, this is what it means. So it's the difference between a channel, a signal, and the reference signal. This is why it's also important for some applications to know which reference channel to use, where to place it, because that may influence also the quality of your signal. We probably are going to have a look at that later. Okay, so this is the very first part of the chain, differential recording. Let's dive a little bit more into the next part of an EEG amplifier, hardware filters. So, after getting the data into the box, into an EEG amplifier, the data pass, the signals pass through one, if not two, filters before reaching the analog to digital converter. You see, one is for sure there. The other one may or may not be there, depending on the hardware, depending on the application as well. The first one that is always there is a low-pass filter or high cutoff filter. Why? Because when we record the signals from the brain, there may be other signals, there may be other noise coming from the environment that is of very high frequency content. And these very high frequency content sometimes cannot be analyzed properly by the analog to digital converter. So with the hardware filters, with the low pass filter, we want to remove those very high frequency content before it arrives at the ADC, before it arrives at the analog to digital converter, so that there's no issue in transforming them from the analog to the digital domain. In the next part, we're going to see why. So this low-pass filter is always there. Some amplifiers may also have a high-pass filter. So a filter that cuts out very low frequencies, maybe the DC, maybe other very low frequencies. Why do I say sometimes it's needed, sometimes it's not needed? For TMS, for example, we don't want that. We want all the frequencies also from DC. So we don't want this filter to be there. We want the DC, we want the low frequencies to be recorded. For other applications, we don't need that. If we are simply sitting in front of a computer and we are doing ERP, maybe we don't want those frequencies, low frequencies. So we can discard them. In general, recording low frequencies is not recommended, but is a good idea, and then you can remove them offline. Whilst removing very high frequencies is very important to prevent problems with the ADC. Just to give you an example, I take here all our amplifiers. So these are all our amplifiers. Some of you may be working with them. Just to give you an idea of what it means. So we have the Actichan Plus, we have the BrainAmp Standard or MR, the BrainAmp DC and MR Plus, and the LiveAmp. And you can see here, these are the high cutoff frequencies. You can see that, for example, the Actichan Plus has a low pass. The high cutoff frequency is at 8 kilohertz, so it's pretty high. Let me go straight to the BrainAmp DC and MR Plus. With the BrainAmp DC and MR Plus, you can choose between 250 or 1 kilohertz, for example. 1 kilohertz is also pretty high. LiveAmp, which is usually used for mobile applications, has a little bit lower high cutoff frequencies. The BrainAmp Standard MR also are fixed 1K or 250. And then you see the high pass, so the low frequency cutoff. There is no low frequency cutoff in the Actichan Plus. You can choose not to have a a high pass with the brain amp DC and MR+, which is important for TMS. Whereas with the brain amp standard, MR, you do have a low pass, a low cutoff, a high pass filter. These are decisions made by the manufacturer when planning, when developing a system, depending on the application for what a specific amplifier is meant for. So these are examples to keep in mind for later. Moving forward, let's then move to the analog to digital converter. So let's imagine we have recorded the signal here. So we have our differential signal that is coming in. We have passed through the hardware filters and we are here now. So the analog to digital converter has two important features. One is the time resolution, one is the amplitude resolution. Let's look at both in more detail. So what the ADC does, the ADC takes a continuous signal in time. It picks in the time domain. So now I'm looking at the x-axis. It takes specific points in time and the relative values of the continuous signal, and it reports them then digitally on the computer. It saves them, okay? So, and you can see that the closer this point in time are, the better the representation of the signal then digitally becomes nicer, of course. So here, for example, this is a representation with a time resolution. Time resolution is the distance between these 2 points, okay? Between two consecutive time points in time. That's the time resolution. This is a time resolution of 0.2 seconds. This is a time resolution of 0.4. Yes, or the other way around. So yes, 0.4, it's wrong. 0.4, 0.2 here, sorry. So long story short, the higher the time resolution, the better the signal is represented digitally. This relates, of course, to the sampling rate. So the sampling rate is the inverse of a time resolution. So the higher the sampling rate, so the more points we have in time, the more points we are picking up from the signal in the time domain, the better the signal gets represented. Okay, so it's more nicely represented. How does this have an impact on our EEG data quality? So depending on the content that you want to analyze, that you want to find from your recording, you may decide to have a specific sampling rate. So by not definition, but by recommendations, you should choose the sampling rate that is at least 5, if not 10 times more the frequency of your signal of interest. So for example, let's say that we want to record ERPs. So ERPs, usually they have a low frequency content. To record up to 50 Hertz. So in order to have a nice recording for ERP analysis, you should go 50 Hertz per 5 per 10. So in between 250 and 500 Hertz. So if you record at 500 Hertz or even 250, you should be good, okay? You should have a nice content for ERPs. Of course, if you want to go higher, of course you can, and you should, because it will give you even better data up to a certain value of sampling rate. What happens instead if we don't have enough sampling rate? So what happens, for example, I decide that I want to do ERP, and my content is up to 50, but I record at 100. I record at 70 Hertz. What does that happen? So let me show you visually first. So here, this is a nice picture, okay, of a brick wall. You see the brick wall is pretty intense, so it's quite intense. If I capture the same brick wall, however, with a camera that does not have enough power, enough resolution, this happens. You see these white things? So something starts to come up in the image that is actually not there. So when the wall is like this. However, I capture the image with a camera that is not powerful enough. So something that is not there, fake content comes up. So this is a moire pattern, but it's a nice visual representation of what happens when you record EEG without enough sampling rate. This phenomenon is called aliasing. So aliasing is the appearance, the emerging of fake content digitally when you're recording something that is not there originally and is due to the fact that we have not enough sampling rate. To translate this into real signals, let's look at this signal. The signal starts slow, so the peaks are slow, so the frequency content in the beginning is not too high, but then the curve starts to be faster and faster. And the dots that you see on the blue line, they are the sampling points. So you see, we are trying to represent in the orange graph, we are trying to digitally represent the curve with fixed sampling points. So the sampling rate is always the same, but the content of my signal increases. Okay, so you see in the beginning, we have a nice representation. So in the beginning, the two signals are correct. However, at some point, the content of my line here, of my curve here, becomes too high and the sampling points cannot keep up. So the curve that is being represented is not real. It's a fake one, it's not the real one. So this is the extreme. So basically, with the fixed sampling rate, if the signal is going too fast, the frequency content is too high, we risk to represent it with something that is not real. Okay, so we basically, in the frequency domain, we add something that is not there, is fake. And in the EEG domain, it means that we may find something in our data and say, oh, I found something, this is a breakthrough, this is a great discovery, but maybe it's simply because we didn't sample the data properly. So it's something that we really should consider, okay? How do we tackle this problem? Okay, how do we solve this? How do we prevent something fake to appear in our data? So let me introduce the concept of the Nyquist frequency. So the Nyquist frequency is, by definition, half of your sampling rate. So let's say I decide that my sampling rate will be 1 kilohertz, so 1000 hertz. Nyquist frequency in that case is 500 hertz. Okay, so it's literally half. And by definition, the Nyquist frequency in an ideal world, in an ideal filters world, is the frequency below which the data can be properly digitized, can be properly recorded and made digital. Above the Nyquist frequency, there will be aliasing. Okay, so ideally, you would think, okay, then if my frequency, my sampling rate is 1 kilohertz, then I am 100% sure that I can record data up to 500 hertz. So content up to 500 hertz, for sure, is going to be nicely recorded. No, not really, okay? Because we don't work with ideal filters. This is where the hardware filters come in. So this is where we introduce the hardware filters that we talked about before to prevent very high frequency content to arrive to the analog to digital converter. So we need the hardware filters to remove very high frequency content. However, hardware filters, they are not ideal filters. So they don't have a step response from 1 to 0. There is literally a step. So they don't stop the frequency immediately at the frequency that we tell them. They take a bit of time to prevent the frequencies to not be passed through. So this is why when we add hardware filters in an amplifier, the high cutoff frequency of the hardware filters is actually before the Nyquist frequency, actually quite before the Nyquist frequency, to prevent that also those parts of the signal that are slowly being attenuated go into an aliasing. They fall into aliasing, okay? So usually, this is why usually when we plan hardware filters, you should have an hardware filter that is around a quarter of your sampling rate. If it's fixed, usually in an amplifier, not just our amplifiers, but in general, in EEG amplifiers, this is a rule of thumb. Or you can have hardware filters that can be with the software that can be switched. So you can, with the software, decide to use one filter on another depending on your application, which is what, for example, happens with the brain amps. So you will see it also in action in the next days. So depending on the application, you can choose which hardware filter cutoff frequency to use. Okay, so this is combining sampling rate and hardware filters, how they can impact the quality of your data. To summarize, I have here also a nice video that shows in theory, okay. This is a chirp signal. It's a signal that, similar to what we saw before, starts with very slow frequency content, and then it goes up to 20 hertz. So it starts from 0, it goes up to 20 hertz. So you see the curve that becomes faster and faster. There is an original signal below. When I play the video, you'll see there is in blue. The orange line is the sampled one, so it's the one that is digitized. The vertical lines are the sampling points in time, okay? So what happens when we decrease the sampling rate a lot? So what happens? Probably at some point we will not have a good representation, a good sampling. And the difference from top to bottom is that the top one doesn't have anti-aliasing filters on. The bottom one has anti-aliasing filters, okay? So what happens is, for example, here we are recording at 100 hertz. 100 hertz is 5 times 20 hertz. So we're good with that, okay? So based on the recommendations. What happens instead if I decrease my sampling rate to 50 hertz, for example? So 50 hertz is 2.5 times the maximum of the frequency content of, of my, of my signal. This thing has a personal life. 75. Okay, let's see if it stays fixed. So you see that the high frequency content starts not to be properly sampled, okay? But the low frequency content is still okay sampled. If I decrease my sampling rate a little bit more, okay, but too much. If I decrease it a little bit more, for example, here I am at 35, okay? At 35, this is definitely not a good sampling rate for this signal, especially for the high frequency content. Why does it work? It goes automatically. What happens is that not just the signal in the low frequency content starts to be not properly sampled, but if I don't have hardware filters, why does it have a personal life? Okay, let me see if it stops here. If I don't have anti-aliasing filters, the content above the Nyquist frequency starts to be picked up, starts to be sampled, but it's not there. It's not real. So you see, the orange line contains a content that is fake. If I have anti-aliasing filters, then slowly there is still a bit of fake content right after the Nyquist frequency, but then it is basically zeroed out. So this to show the importance not just of having the right sampling rate, but also having the right sampling rate paired with the right anti-aliasing filters. Okay? At the end of the whole part, there will be an association of these features to the TMS application. So you'll see why it's important, like tailored to the TMS-EEG application. Okay, so this is all about time resolution, sampling rate, and anti-aliasing filters. So we're talking about the time domain. And here you can see, as examples always, the sampling rates for our amplifiers. So, and these are hardware sampling rates. So the analog to digital converters, they have fixed hardware sampling rates. And then with the software, you can choose a different sampling rate. So the software will have additional steps, decimation, an additional anti-aliasing software filter, if you want to use a different sampling rate, okay? But the fixed sampling rate hardware is, for example, for the Actichamp Plus, there are 3, so you can choose 10, 50, and 100 kilohertz, very high. The brain amps, they have a fixed 5 kilohertz hardware sampling rate, but this doesn't mean that you cannot use 1K, you cannot use 500 hertz. Yes, of course you can, and then the software will take care of adjusting all the other anti-aliasing filters and so on accordingly. Now, let's move from the time domain to the amplitude domain, because the ADC samples the data, of course in time, but also in the voltage, in the amplitude domain, with the same principle, as similar, okay? So here we are not looking at this x-axis, we are looking at the amplitude, so the y-axis. Same concept, so the ADC will sample the continuous line with specific points, at specific points in the y, on the y-axis. And same principle, the more points we have, so the thinner these steps are, the better the signal is sampled, the better the signal is represented. So exact same concept. Here we have to introduce another principle, the principle of the bits. So the bit for any digital device, a computer, an EEG amplifier, anything, is the smallest unit of data that the device can record, that the device can process. And usually, EEG amplifiers, computers, and all the devices, they come with a technical specification of bits per volt. So you may have heard this device, this EEG amplifier has 16 bits per volt, 24 bits per volt, also with the computers. What does that mean? So the more bits per volt a device has, the higher the amplitude resolution it can have. Why? So given the number of bits that a device has, let's imagine 16 or 24, it implies that it will have a specific number of possible steps, of possible points on the y-axis that can be sampled, okay? So let's imagine we have 2 devices. One has 16 bits and one has 24 bits. The equation, the formula is calculated 2 at the power of bits. So here we have with 16 bits, 2 at the power of 16, this device with 16 bits will have 65,000 possible samples here on the y-axis. If we have 24 bits, we have actually 16 million possible steps, which means that they are probably thinner. They are probably, it's more accurate. It's going to be more accurate. Given the bit depth, which is this total number here, and given a specific resolution that we want to have here, so a specific difference between one value and the other, we can talk about measurement range. what is the measurement range? The measurement range is the maximum and the minimum value, voltage value, that a device can record, that a device can represent. How is that calculated? Imagine we have a device with 16 bits, which means that it has possible 65,000 values. And I decide that I want a resolution that is very, very thin, that is very, very small, of 0.1 microvolt per bit, which means that this height here, this step here, I want it to be 0.1, okay? I want it very small. That means that, okay, 0.1 per 65,000, that means the possible values is around 6 microvolts, 6,000 microvolts. Then I turn into millivolts, and I have to go plus and minus, because EEG plus and minus. So I have a measurement range plus minus 3 something millivolt, okay? So imagine an EEG amplifier with these features, so 16 bits with a resolution of 0.1. The measurement range, so the volt values that it can properly sample, that it can properly depict, are between 3 plus and minus 3 millivolts. And you say, well, the EEG is very small, so it's in the microvolts, so it's not a problem. Yes, true. However, when working in specific applications like with TMS, something else is coming into the recording, like a TMS stimulus, which is going to let the EEG recording skyrocket. So what happens if the amplitude resolution is not enough? What happens is that we risk this effect, the clipping effect. So if the amplitude resolution is not the amplitude resolution, if the measurement range is not enough, and our signal goes too high or too low, we risk that it's clipped out, that it's cut out. So we are missing some information. Furthermore, and we'll see it in a bit, if this clipping lasts for a long time and the signal that goes out of range is really high, the amplifier may go into saturation as well. So the amplifier may saturate, and it will take a little bit of time before it goes back to a proper recording, okay? So this is something also, especially in combination with TMS, that should be kept into consideration. And let me give you an example. So the brain amp that you will use in the next days, they have the possibility of choosing your resolution, your amplitude resolution. Why? Because the brain amps have 16 bits. So they don't have 24 bits, they have only 16 for a specific reason. But you can choose between two different amplitude resolutions. So if you choose the 0.1 microvolt per bit resolution, you will have quite a small measurement range, plus minus 3 millivolt. But if you say, okay, I actually do not need such a low amplitude resolution, I can live with something that is a little bit bigger, like 0.5, then always if you do the formula that we did before, your measurement range increases. So you give up a little bit on your resolution, on your amplitude resolution, but you increase the measurement range and you prevent the clipping effect, you prevent the possible saturation that is important for certain applications like with TMS. Okay? Now, we're almost, a summary. So what we just said, we talked about both the time and the amplitude domain, and we said the higher the sampling rate, so the higher the time resolution, the higher the amplitude resolution, the better. Yes, up to a certain point. So there is a trade-off between how high the sampling rate, how the amplitude resolution is. Because you can go really, for example, with the Actichem Plus, you can go up to 100 kilohertz, which is really, really high. However, at some point, two things. First of all, physiologically, at some point, there's nothing else to record. So what you record with such a high sampling rate is much more noise. So you will see that the line at some point becomes super fast. Thin but super fast is because the noise that is around is being picked up very nicely. So at some point there is not much to go above with the sampling rate. And more or less, furthermore, if you record with such a high sampling rate, your data, your files will be really heavy. So it will be giga and gigabytes of data that not just your computer probably will struggle at some point to save, but the analysis software that you use will struggle to import and analyze. So there is a trade-off at some point that you have to choose on which is the right sampling rate, high enough for what you want to study, what you want to find, and also the limits that it will have physically on your computer. So very high sampling rate, very high amplitude resolution doesn't mean there is the best. There's always a trade-off. Okay. As an example again, before I move on, measurement ranges. The Actichan Plus has a pretty high measurement range, plus or minus 400 millivolt, more or less. With the brain amp DC MR Plus, you can choose, as I said, you can choose between 3.28 or 16, whatever, millivolt. And spoiler, for the TMS, of course, you should use the biggest measurement range. To continue, last two important features, amplifier and electrode impedances. So this is something that if you have worked with EEG, is one of the first things that you do when you prepare a participant, lowering the impedances. Before talking about lowering the impedances, let's have a look at what this impedance concept means. So when we record EEG data, we have the brain with the signal inside the skull, so we have the brain. And then the signal travels through many layers. There's the subcutaneous layers, there's the skin, there's the ears, and then there's the gel, and then there's the electrode, and then finally it goes into the EEG amplifier. So there's quite a few steps that the signal has to go through. And especially these parts here, these layers between the brain and the amplifier, they can be represented by a system of resistances and capacities. Okay, there are quite a few studies that tried to simulate with electronic circuitries how these actually behave. What does it mean? It means that the signal goes through a lot of layers before being picked up by the electrodes. And it is important to have a good impedance, not just with the electrodes, but also with the amplifiers, to be able to pick up good signal, sorry, and leave out the noise. The best signal is recorded with not just low electrode impedance, but also with high amplifier impedance. So the combination of these two is what makes the signal very nice. This is something maybe that you don't hear that often, because the EEG amplifiers, not just ours, but also all the other manufacturers nowadays, they all have very high input impedance. So if I have to give you the examples from our amplifiers, you see that the input impedance of the amplifier is very high. So it's in the ranges of megaohms, if not gigaohms. And also other manufacturers, they all have very high input impedance, because it's really important to have that in combination with low electrode impedance. However, using amplifiers with very high input impedance allows us, for certain applications, allows the research world for certain applications to use also different types of electrode technologies. So Of course, these are examples, again, from our portfolio, from our electrodes. Probably many of you, especially if you do TMS, you have worked with the passive electrodes. Okay, so these type of electrodes, they have by usually very low input impedance, but there are also other type of electrodes, like the Arnets, for example, with the sponges, or the dry. By default, they have higher electrode impedance because they follow a different technology. However, since we use very high input impedance with the amplifiers, we are safe in using these type of electrodes for certain applications, okay? Not for all applications, but for certain applications, like recording EEG in a static position at the computer in a lab. You're safe because the amplifiers will sustain signal that is a little bit more noisy, okay? But for TMS-EEG, of course, we want the best type of signal quality, so low electrode impedance is important. And how do we get there? With a good skin preparation, okay? So the bare skin has a very high impedance, okay? The bare skin. Protected skin has a little bit lower impedance, and preparing it allows to reduce the impedance value. Depending on the electrode technology, with passive electrodes, for example, we are all used to abrade the skin also, so to really scratch the skin a little bit in order to get away a little bit of sweat, get away a little bit of dirt there is there. So this is the reason why we abrade, okay, to lower the impedance value of our skin. It may not always be needed. For example, if you use active electrodes, the active electrodes are capable of amplifying the signal, making it look nicer. Let me say it in these very lame terms. So you don't need to abrade the skin, and you also use a different type of gel. So depending on the electrode technology that you use, you may prefer one gel or another, okay? With passive electrodes, usually you have to abrade the skin, so you would use an abrasive gel. Active electrodes, you don't need to abrade the skin. The active circuitry will take care of making the signal look nice. So you can use a gel that is a little bit more viscous, more liquid. Okay. Now, okay. Okay. So this was all in terms of getting to know the EEG hardware, okay? So we talked about all of these important features. So we now should know how an EEG amplifier works. Let's try to translate this into the TMS-EEG, the simultaneous TMS-EEG application, okay? So when we do EEG, when we record EEG alone, what do we want to do? So we want to have a nice environment that is kind of protected by external noise. We try to ask the participant to relax, not to tense the muscles, not to move too much, so that the data look nice. So maybe we even work with the Faraday cages to prevent any strong interference to come in. So this an ideal EEG recording. On the other side, on the TMS side, if we do only TMS, okay, ideally we try to place the coil very, very close to the target, so we really attach it to the scalp, and we don't want to have anything underneath the coil that may prevent the stimulation to pass through properly, okay. So when you think about this, and you think about, okay, I want to do simultaneous TMS-EEG, so at the same time, you see that there is something that is not really working here. So the two ideal worlds are conflicting. So we have conflicting requirements. So there are different ways in which TMS-EEG can be made, and it is made. And this, of course, comes with also what Henri mentioned before on how TMS works. So the TMS coil induces an electromagnetic field, magnetic and electrical fields that are really, really strong. So the intensity is expressed in Tesla, and it goes up to 4 Tesla. And I believe, depending on the TMS manufacturer, then the strength of the pulse can be different. The frequency content of the pulse is also very, very high. So the pulse is very short. It's like it's very, very short. It doesn't last long in time, super, super short. So the frequency content is high. And the effect that it has on the brain depends on the shape of the coil. So usually we have all most likely worked with the butterfly coils, because those are the ones that are more precise, right? Precise is the right definition for the coil. They point to the right spot in theory. So depending on how your coil is, it may affect the brain in a different way. And there are 2 different types of TMS-EEG combinations. There are the offline combinations, for example, where you stimulate, or actually, well, you record the EEG first, then you stimulate, and you record the EEG again. This is, I would say, the safest of the options, because you're not doing anything simultaneously. You're just recording EEG, then applying the TMS, and maybe this is done also on a different moment of the day, on a different moment of a week, of a month, okay, because you want to monitor pre and post. It's easy. The only thing is that here you're gonna look at what happens at the brain in the long term. So you don't know what is really happening in the brain when you stimulate, okay? So you see only the after effects of a TMS intervention. This is why the online combination, the simultaneous TMS-EEG, is so interesting, because if you can technically make it work, you can actually see what happens in the brain right at the moment of stimulation. Maybe not right at the moment, but just a few milliseconds after the stimulation. However, it is of course challenging because of the conflicting requirements of the 2 worlds, of EEG alone and TMS alone. But this is what we want to learn a little bit more today, and what you're here to learn more in the next days. Okay, so this is the application that we want to learn a bit more. Now, try to remember what we just talked about, talking about the EEG features, the hardware features, and let's try to relate them to a TMS-EEG study, a TMS-EEG recording. There are some artifacts that you will always have, some always have, some maybe not always, so ideally never. That are related to the EEG hardware and the fact that it's being used together with the TMS. The first one is the ringing artifact. The ringing artifact comes from the very high frequency content that a TMS pulse has. So as I said, the TMS pulse is very short, and usually we would say that the frequency content is up to 3 kilohertz. Okay, so imagine I'm recording EEG, and then another signal comes in, that is the TMS, which has a very high frequency content, 3 kilohertz. And if you remember what we just said before about sampling rate and about anti-aliasing filter, what do you think will happen if I don't have enough sampling rate, or if I don't have enough, so my hardware anti-aliasing filters are too low? What happens is that you get this type of red response. Okay, so the pulse is very fast, but since I don't have enough sampling rate, plus my hardware filters maybe have a very low cutoff frequency, the very fast line, the very fast pulse of the TMS gets spread out very long in time, and this is actually not nice. This is bad, because this is going to cover, to hide real content, real EEG content. So what is actually a very fast, maybe a very fast like line, a very fast deviation from the 0 line, that could be 1 or 2 milliseconds long, gets spread out in a way too long period of time, and you lose a lot of data, okay, useful data. Okay. This is called the ringing artifact, because it rings up and down. So that's why it's called like this. How do we prevent this from happening? We try to work with amplifiers that have a high input, a high cutoff frequency in the low pass input filters. Okay, at least 1 kilohertz, at least. If not higher, higher would be even better. And then we try also to work with a high sampling rate. For TMS-EEG, 5 kilohertz is a good sampling rate. You can also go up a little bit more. For example, with 25 kilohertz, that is possible with the Actichan Plus. At some point, the ringing artifact is not there anymore, okay? Because with 25 kilohertz, you have quite a lot of points in time that you can sample. So if you remember how the signals were sampled, you don't simply pick points not so close to each other. You're actually representing it very nicely, okay? So this is the effect of choosing an amplifier that has enough sampling, an enough high cutoff frequency with the low pass filter and a good sampling rate. Okay. Next. Amplifier saturation. So we talked about this when we talked about the amplitude resolution, the measurement range. This is due to the fact that the TMS pulse is very strong. So it has a very big volt to volt difference. So this is a normal EEG curve. So the peak to peak of a normal EEG curve. This is the peak to peak when a TMS pulse comes in. I always like to compare it to the difference between the Shanghai Tower and the height of a coin. Okay, so the impact is pretty strong. And what happens, what may happen if your amplifier does not have enough measurement range, we said that before, you may risk to have a clipping effect, okay? So you may lose some data, not just, but the amplifier may saturate. So there's the TMS pulse, and the TMS pulse is so strong that the amplifier cannot keep up. Simply it saturates and it loses a lot of data until it recovers, until it discharges and is capable of recording EEG again, okay? So let me phrase it that it was a problem because in the past, technology, EEG technology 20 years back, not just ours, but any manufacturer, the solution that was followed in the past was a technique called the sample and hold. So at the moment of stimulating with the TMS, the amplifier would pause for a moment, for a few milliseconds, in order not to be affected by the TMS pulse, and then it was starting to record EEG data again. However, this is a very old and obsolete technique. Amplifiers any amplifier nowadays doesn't work like that anymore because of different reasons. So first of all, you're losing data because you're pausing your TMS, your EEG recording for a few milliseconds. So you're actually losing some data that maybe you'd actually be able to analyze afterwards. The TMS artifact, the TMS pulse, also influences the brain in different ways. So if I stimulate the motor cortex, the channels that are on the occipital or the frontal area are not going to have the same TMS pulse. So maybe there's no need of stopping the recording there. But in the past, when you were doing sample and hold, all the channels were off. Okay, so it's a technique that has been obsolete since a while. What do we work with? Amplifiers with big measurement range. So with a big, at least tens of millivolts peak-to-peak volt range. Yes, range. Right. Then, last but not least, this is the nightmare of any TMS-EEG researcher, the decay artifact. The decay artifact also is related to the hardware, but is related to the preparation of your participant, the type of electrodes that you're using, and the fact that, as we said before, between the brain and the amplifier, there's a lot of layers. They can be represented by a series of resistors and capacities. What does it mean? That when we give a TMS pulse that induces a current, all these capacities will charge, okay? So they get charged, and then they slowly need to discharge. So this is the decay artifact, is the fact that these layers get charged a little bit and they have to discharge slowly. So the goal is to make sure that these capacities, this series of capacitors and resistors gets charged as little as possible, okay? How do we do that? The risk is that if we don't do this, the decay artifact may be very long. It will go up to 20, 40, 50 milliseconds, so you're losing a lot of data. The artifact is covering real data, okay? How do we try to solve this? So first of all, we should use TMS-compatible electrodes, okay? I wouldn't use dry electrodes, for example, in a simultaneous TMS-EEG experiment, okay? There are some electrodes that are a no-go, only TMS-compatible electrodes. You need to work with a very high amplifier input impedance, like we said before, as well as a low electrode impedance. And so if for certain applications you can work with a higher impedance, like for example using active electrodes with 25 kilo ohm, with the TMS, when doing simultaneous TMS-EEG, we would still recommend going low, so going below 10 kilo ohm, for example, regardless of passive and active. And another tip is trying to optimize your cable routing. So this is a very nice study by Sekiguchi and et al. They tested the different orientations of the coil and how this influences the decay artifact, and basically stimulating the coil perpendicular to the cables, the electrode cables, is the position that minimizes the decay artifact in an ideal world, of course. So if the electrodes are TMS compatible, if the impedance is low, so in an ideal world, that's the position that is optimizable. Last but not least, another tip is to move the ground and the reference electrodes away from the TMS spot. So in the next days, when you do the hands-on sessions, you will see good positions where to put the electrodes. This is another thing to keep in mind, because if the ground and the reference are really under the coil, well, there's going to be a lot of noise in all the electrodes. Okay? I think this is it. All right, we have a quiz time. Let's see how much you have learned so far. So I'm gonna ask different features. Different features will pop up. I would like somebody from the audience to answer which artifacts it may be related to. It's gonna come up also online, so there will be a poll online and you can answer the poll. Okay, let's see what comes up first. Okay. Well, this is easy, we just talked about it. Okay, skin and gel preparation, what does it affect? Don't be shy, I don't bite, I don't judge. Right? Anybody? Go for it. Liam? We have the cubes.
One.
The decay artifact?
Right, perfect. Nice, okay. Then, what comes up next? Hardware filters. What do they influence? Come on, guys, don't be shy. I know you know. One hand raised. Yes, thank you.
Where?
Front of you.
Thomas. So the ringing artifact.
Yes, thank you. Yes, all right. Amplitude, resolution, and measurement range. What does that influence? I'm getting scared that I was not clear enough now. Oh, two hands.
The saturation?
Yes, very well. Okay, we're almost there. Amplifier and electrode impedance. Nobody? Really? How is the online going? What do they say online? I don't hear you. Saturation. Come on, guys. Amplifier and electrode impedances influence decay artifact. Oi, oi. Fortunately, this is going to be recorded, right? So you can rewatch it.
Watch on YouTube again.
Yes, perfect. Last one, time resolution and sampling rate. Easy? Anybody? Ringing, great, perfect. Nice, okay. This is a nice summary of EEG hardware features and how they can influence TMS-related artifacts, okay? Always to keep in mind. And this is a nice summary, again, just as an example, but if you're using other manufacturer, it's nice also for you as an exercise to go on the technical specifications and check your EEG amplifier, how it works, okay? It's actually a good exercise. Okay, as a summary, we said, okay, we want an amplifier that has very high cutoff frequency in the low pass filters. So, for example, the Actichamp Plus and the BrainAmp DCMR, they have 8,000, 1 kilohertz high cutoff frequency, so that looks good, as well as DC option. Okay, so we're good in terms of being capable of detecting the ringing artifacts in a good way. The hardware sampling rate is also pretty high, still for the ringing artifact. Measurement range is also good enough. Well, the Actichamp Plus has a very big measurement range. The BrainAmp DCMR Plus, you can choose and go at 60 millivolts, so good enough as well. Amplitude resolution also is good for both. So these two, if I were to talk about my portfolio, these two are the two options that I would recommend to somebody that wants to do TMS-EEG. Maybe a disclaimer that the BrainAmp MR Plus, especially, they had been designed to work with DMR. So they can be put into the MR scanner. Therefore, the hardware filters that they have, they are particularly suitable to work against the magnetic interferences. So this is why the BrainAmp DC and the BrainAmp MR, they are very well suited for TMS-EEG as well, despite maybe the fact that they have a little bit lower other features. Still, they work very, very well, okay? So knowing your hardware is always important. I have a couple of more slides because we always talk of how the EEG hardware may influence, am I going? Well, 1 minute. Okay, I'm almost done. We always talk about how the EEG hardware influences the data quality, but TMS also can also influence the data quality. How? Recharge artifacts. So when we stimulate with the coil, the coil gets discharged and it needs to be recharged. And this comes up as a tiny stimulus in the data, a little bit later after the real stimulus, but If this is not late enough and it's not recognized, it may be analyzed and it may be mistaken for a real EEG data. So it is important to, with the TMS nowadays, it is possible to delay the recharge time in a way that it goes after your window of interest, so you're not mistaking it. If you see that it is there, it's important to filter it out, to cut it out, so that you're not mistaking it for a real EEG component, okay? Last but not least, nowadays, I would say no TMS manufacturer has this issue anymore, but new machines, new pilot test systems, they may also introduce a lot of noise in the EEG. And this happens when, for example, the power inside the TMS, the power line is not properly isolated, or when you use coils that have the cooling system really underneath the coil, for example. So this huge artifact, 50 Hertz artifact, is basically destroying your data. So it is important to prefer a certain type of coils as well as to pilot the setup always to make sure that you don't have such a strong artifact in your data. Okay. So to summarize, these are all the most important artifacts that there are when that might happen that you will see when doing TMS-EEG, together with their solutions. Let's call it like this. Maybe last but not least, will there be any other artifacts in your data? Most likely yes, but these are less hardware related. They are more physiological. There may be artifacts that are coming because of your participant or because of how the setup is being prepared. But this is something that Silvia will talk about extensively in the afternoon. Questions? Do we have time for questions? Yes, one question. One question.
Thank you very much, Sara, for this very comprehensive talk. And so please, Liam, if there are questions.
It's an online question about the advice on ground and reference when performing TMS-EEG. Are you suggesting you use a single electrode as a reference and not the mean of recordings in all the electrodes?
I answer, but then maybe you answer. But yes, definitely you use one single electrode as a reference. Then I'm talking to the camera. One single electrode as a reference and placed far away from the TMS coil. So if I remember correctly, you place them on the forehead sometimes, which should be far away from the coil. But it depends on the TMS protocol, I would say. Still one reference electrode far away from the coil, yes. And probably during the afternoon, maybe you will also mention a little bit more about this, yes.
You might have the opportunity to see where we put the reference and ground electrodes. There can be different choices, but yes, most important thing is to put them far away from the stimulation spot.
Traumatic brain injury, not necessarily involving disorder of the conscience, but impairment, motor cognitive impairment. So I said TMS-EEG is unique, and when the rationale for using TMS-EEG in disorders of conscience will become clearer, hopefully, in the next 15 minutes. But what I want to say and remind is that TMS-EEG is really, really unique in many respects. So, first of all, it's a direct cortical perturbation and direct cortical recordings. And when we analyze what happens right under the coil, we have a unique assessment of the input-output properties of cortical neurons, which is very unique. So, stimulating and recording from cortical neurons has been traditionally the way to study neurons and circuits in vitro, sometimes in vivo, in animal models, but this can be done in humans with TMS-EEG, input-output properties of local circuits. Then there is another aspect. TMS-EEG is unique because it's a direct cortical perturbation. And by activating one cortical modules and then by recording with EEG the propagation of this activation, you have a network assessment by causal perspective. This is not temporal correlations, it's causal. It is technically called effective connectivity. This is a very precious way of assessing circuits and neuronal interactions because it's not confounded by common drivers and by other sources of spurious connectivity. So that's a very interesting fact. And then more specifically for disorders of consciousness, but not only, not limited to that, is the fact that TMS-EEG is not dependent on sensory inputs because you bypass sensory inputs. By definition, you stimulate the cortex, meaning you might bypass lesions in afferent pathways, thalamic gating, and you access directly the thalamocortical system from the top. The other aspect is that it's obviously independent of motor output. So we are not measuring EMG. We are not observing movements. We are recording from the cortex itself. So it's an assessment of excitability, connectivity, independent of sensory inputs and motor output, unlike sensory evoked potentials and motor evoked potentials or behavior itself. Last but not least, super relevant for disorder consciousness, TMS-EEG does engage circuits in complex ways without requiring the engagement of the subject in a cognitive task. That is, it's independent of executive functions. And sometimes in patients, executive functions are impaired. And so the patient is not engaging in the task and you cannot read out the cellular and network mechanism just because you cannot engage him in the task. So that's a very interesting thing. Again, relevant for disorder of consciousness, but not limited to that. So why is this relevant for the assessment of patients who emerge from coma, open their eyes, and remain unresponsive, or they are minimal responsive? This is the challenge. To show why this is relevant, I will have to go through, a little bit of, like, not so much in history, but sort of a structured, layered account of how we assess consciousness in patient. And I want to say right away that assessing consciousness patient is like peeling an onion. You have different levels. You have the easy levels, that is the behavioral assessment, and then you go deeper and deeper and deeper. And at the deepest level is where TMS-EEG come into play, as I will show. If you're interested in these levels, or if you are curious about, the nosology and the problems associated with detecting consciousness after coma, these are a couple of references, but I will explain you the layers one by one. Of course, the first layer is overt behavior. That's, the gold standard. All coma scales, coma recovery scales are based on this. You probe the subject with sensory inputs, verbal commands, and then you observe motor behavior. Basically, you treat the brain as a black box, but you work through this input and output directly. And of course, if a patient shows responses that are purposeful, reproducible, you have a consciousness detection, no problem. It can be at different levels, so the patient can be impaired to the point that it cannot communicate reliably with you, but is responding, and there are so many different ways. One key distinction at this level is the distinction between the vegetative state. Vegetative state are patients who are eyes open but fully unresponsive. They show only automatic behavior. And the minimally conscious state, that is a situation where the patient is actually reacting to stimuli. There is an MCS minus, an MCS plus, depending on the complexity of the reactions, but that is a state of consciousness. And it makes a difference because there is a better prognosis, and of course, it's a key difference because the patient is there somehow, okay? So that's a key distinction, and if you see purposeful behavior and responses at this level, you are confident in saying, okay, the patient is more than vegetative, is in a minimally conscious state. However, this level of assessment requires the integrity of so many systems. So sensory input, sensory pathways, they must be in place and in working order. Sensory processing in cortex, primary cortices, they must be in working order. Executive function must be there in order for the subject to provide the motor output, which happens if motor systems are in place. So all the chain must be intact. And so when you see a response, as I said, no problem. But when you don't see anything, the absence of the proof is not proof of the absence, because you could have lesions at levels that are not necessary for consciousness, but they are impairing your assessment. One example, for example, let me just show you this, how the scales are working. So you have this different situation. Coma is eyes closed, eyes opening is vegetative state. And then by looking at motor behavior, response to command, you have this interesting stratification. MCS minus, purposeful movement, MCS plus, they follow command, squeeze your hand. Confusional state, patient is there, but is confused, and then you have different levels of cognitive dysfunction. But we need to remember that this key distinction here between unconscious and conscious is drawn based on an inference that is based on motor behavior. And if you have lesions to motor system, then you won't see much. And indeed, we all agree, also in anesthesiology, that unresponsiveness does not mean necessarily unconscious. Indeed, you might have subjects who are perfectly conscious, but they're blocked on the motor output. Locked-in patients being an example. I don't know if you know what is a locked-in syndrome. Is a situation where a lesion in the brainstem, in the ventral part of the pons, is blocking all the outputs. And in that case, your behavioral assessment would be a false negative. Patient is conscious, but it's just fully paralyzed. So here comes the next layer, because we know that the sensitivity of this behavioral scale is not great. So we have an estimated rate of 30–40% false negatives. That is, patients who are wrongly diagnosed as vegetative. So of course, the community has gone deeper. And over the years, methods have been developed to go beyond this level, and specifically to go beyond the motor impairment. One very smart way to do so is to elicit activations by giving the subject verbal commands, but then rather than looking at movements directly, you look at neuron activation upstream to the movement. So this has been done in fMRI first, and there is this very famous paper, I don't know if you're familiar with, it's now almost 20 years old, the paper, where if you put healthy subjects in a scanner and you ask them to imagine to play tennis, you see this clear-cut activation in supplementary motor cortex. If you ask the same subject, healthy subject I'm talking, okay, don't move, just imagine that you are navigating in your own apartment, you have a different set of activation, a different constellation, including memory systems and areas that are involved in spatial coordinates, as well as motor systems. And so you know that the subject understood you and willfully did something, which is as good as a motor response, but without a motor response. So this work by Adrian Owen, at the time he was in Cambridge, revealed that some vegetative state patients, they can do the same, meaning that they are conscious. So it's a work that fundamentally confirmed the notion that the behavioral assessment lacks sensitivity. This has been now validated. There was a recent review on the New England Journal of Medicine, multicentric study showing that this works, that you can detect 25% of vegetative state patients, they have actually this capability. They are called cognitive motor dissociation, which explains very well, describes a situation where all cognitive processes are in place, except for the last bit of providing a motor output. However, also this layer implies the integrity of so many systems. So the subject needs to understand the verbal command, needs to be able to process it and to concentrate, because it's not easy to produce this activation. You need to concentrate and really imagine to play tennis. Let's say you're a patient, you're in pain, you're confused, you don't care. So there are so many reasons why you can fail at this level, including problem with executive functions, because this is really implying executive functions. You want to do this, you want to concentrate, it requires a lot of attention. It's very demanding on cognitive functions in general. And so what we know, that also here we have a high false negative rate, about 65%. It means that patients who are behaviorally conscious, like minimally conscious or more, they fail. And sometimes also healthy subject can fail. So this is another level. It can compensate for some errors at the behavioral evaluation, but it's not the solution because of this high rate of false negatives, because it's very demanding on cognitive executive function. Now come the third layer. We are peeling the onion. So what if a patient is negative here? Well, it could be a false negative. Then it's also negative here. Well, it could still be a false negative. Then you need to dig deeper. And the next level is a level that implies stimulation in a more passive sense. So what you do here is quite complicated in the sense that you provide the subject with stimuli of maybe growing intensity, complexity, and then you look at the cortical activations. And in general, we think that cortical activations that are selective for more complex stimulants, that are more widespread, are a marker of conscious cognitive processes. There's a lot of debate here. So this is a difficult layer because we haven't pinned down the neuronal correlates of conscious perception. So we don't know whether a given neuron activation is for sure a sign of consciousness or not. There's a lot of debate. This is where the conscious community is getting, really into hot disputes and also a little bit stuck. So it works, but it's, as I said, quite challenging. One example is the so-called local global paradigm. This is a noteball sensory stimulation where you have frequent stimuli and then sequences of complex stimuli. The evidence is that you need to pay attention to this, and if you do, and if you're conscious of this deviant, you have a specific waveform in the EEG now, not fMRI, that is called the P3B. Have you heard about the P3B? It's a wave that is late and positive, which reflects high-level cognitive processing of deviant stimuli. And it's taken as a signal of consciousness. So when you see a P3B in a patient who's otherwise negative here, well, you have to question the diagnosis because something is there, okay? Quite specific. However, also in this case, sensitivity is very low. Actually, it is pretty low. And this is because the P3B still requires some level of executive function, frontal lobe function, such as attention, to the point that you have positive P3B only in 30% MCS patients, so that would be a sensitivity of 70 or 69%, which is not great, and a sensitivity in conscious subject like me of 52%. So again, this can help, can detect some false positive at previous layers, but still you have a residual significant number of false negatives. And now the last level, that is the final last resort of having to look to things that are really into the brain. Also because this sensory perceptual activation can fail simply because you have damage to sensory inputs, for example. So the fourth layer. The fourth layer is a layer where you are not assessing any perceptual cognitive function. You deal with an object. This object is the brain, and you have to make an inference based on internal brain processes. That is fundamentally about physical processes in the brain. It's like pointing a radar on a structure or on a planet and saying, is there life in there? Based on, you know, validation, redeploying things that have been validated, and making an inference. Well, assessing consciousness, it's always an inference, but this is an extreme inference because it's based on physical properties. But it's a necessary step. So this is just to say that we cannot just be happy with this, not be happy with this. This is not enough. We need to dig deeper. If we want to detect consciousness, that's what we need to do, although it is challenging. This is because there are cases where the brain, after injury, is temporarily or maybe also permanently completely disconnected from the external world. And this can happen in many ways, both on the input and the output side. You can have frontal lesions impairing executive functions. I will show you a case. You can have lesions, for example, in the left language networks, impairing the ability of subject to understand commands. You can also have lesions in the brainstem, impairing input and output pathways, and all combinations of this. Diffuse axonal injury. There are plenty of examples. There are so many ways you can become disconnected. And when this happens, and when you want to assess consciousness in this case, well, you have to deal with measures, develop measures that are based on the brain, that are independent of sensory processing, executive function, motor function. So basically this layer. And now you see why TMS-EEG is a useful tool, independent of sensory processing, motor output, executive function, but still provides informative indices of thalamocortical function. So this is the level that we are exploring. At this level, there's not only TMS-EEG. Of course, there is EEG as well. EEG is a great tool without TMS. So EEG is something that has been discovered, as you know, about 100 years ago. And already Berger, the pioneer, realized that EEG waves, they have a relationship to vigilance and the state of consciousness. A slower EEG with big waves in the delta range is, for example, the epitome of deep sleep, where we are mostly unconscious. It is present in many types of general anesthesia and coma as well. So looking at EEG, it's a good thing. And for example, you know, we are in the age of AI, multivariate measures and all this. There's an interesting paper recently combining all the measures, including fMRI, EEG, multivariate AI-based kind of, you know, vector machine learning. And it turns out, that's a very interesting finding, I think, a little bit nested in the tables of this paper, that the EEG is still the best thing. Looking at the EEG by a clinical point of view, looking at the waves, okay, better than any other automatic machine learning index. Indeed, we know that if you have an EEG that is preserved, that is, you have, for example, alpha in the back, you have an anteroposterior gradient, you don't have a lot of delta, it is very likely that the patient is conscious. Whatever the diagnosis at the previous 3 level, a good EEG should cast a doubt on your diagnosis. On the other hand, an EEG that is slow or suppressed is not a good sign, so it would confirm by an intrinsic property perspective of the brain the previous negative diagnosis. However, there is a very significant large gray area in the EEG domain. So there are many EEG patterns that are inconclusive. Slowing, diffuse delta, theta, it's unclear. So here, the EEG does not provide definite answer. And this is because, by the way, it's been recently described, what we sort of knew, that you can have consciousness also in situations when you have a lot of delta waves at the EEG level. This is because the EEG level is picking up because of volume conductions, delta foci in a very, they become very prominent at the scalp, and they can mask maybe activations in the brain that are relevant for consciousness, but you don't see in the EEG, for example. So again, within this fourth level, we need to dig deeper and go beyond EEG rhythms per se, and that's where finally TMS-EEG comes into play. And it comes into play in an interesting way. Forgive me, but I have to start with a little bit, of a theoretical rationale for using TMS-EEG. This is already good practical rationale, we're bypassing sensory input, motor outputs, but what do we measure then in the brain? What is relevant for consciousness in the physical brain? Well, there is an idea that is related to complexity, network complexity in thalamocortical circuits. The idea goes back to more than 20 years ago, 98. This was a paper by Giulio Tononi and Gerard Edelman. That was the first paper suggesting that if you want to assess consciousness, so at the time, in the 90s, synchrony in the gamma band was the main neural correlates of consciousness. But then they said, no, look, it's not so much, the EEG, the ongoing EEG, or the specific spectral features. It's some kind of specific complexity in the brain. The idea is very interesting because it starts from an observation of the properties of consciousness, so phenomenological properties. That is, the idea is very simple. Consciousness is very rich and unitary. So if you want to make an inference on physical properties, you have to look into the physical object for richness and unity at the same time. Technically speaking, it's the coexistence of integration and differentiation in thalamocortical circuits, this balance between unity and diversity. So more operationally, the idea at the time was, this is taken from the paper directly, so integration matters. Group of neurons contribute to consciousness to the extent that they are interacting so strongly that they form a whole, one object. And differentiation instead is the idea that this same group of neurons forming a whole, it also needs to be capable of sustaining a large repertoire of states. And this fits with this unity and diversity that characterize conscious experience. So this was basically in a nutshell the idea. Of course, one should give a talk of 2 hours just to discuss this. But the idea then is that what you need to measure is this coexistence of differentiation and integration. Very difficult, not only to achieve in systems in general, societies between segregation, globalization, in political systems between, if you want, anarchy and dictatorship. But in physical system, it's very difficult because these two forces are working one against the other. The more a system is differentiated, the less its communication is more difficult. If you make it, tightly connected, then you might lose differentiation and this diversity collapses. So it's a very delicate balance. It's also difficult to measure. And it's a little bit metaphorically speaking like, an orchestra where you have different elements with different partitions and they all work together. So this idea, we reviewed it with Simone, who's going to be also in the team that will, be teaching in these days. It's an idea that was put forward 20 years ago. The field of consciousness is very much controversial. I don't know if you heard about, there's a lot of controversies. Of course, it's a hot and debated topic, but on this idea of complexity, there is a lot of agreement. So we reviewed the literature. Indeed, there are so many papers that are confirming that measures of complexity are useful. There is a growing literature that with a delay, as soon as tools became available, is picking up, is growing exponentially. There are attempts measuring complexity in different situations, such as anesthesia with fMRI. in patients, and it's an interesting field. So many techniques and different cases of loss and recovery of consciousness, including seizures, sleep, anesthesia, disorder consciousness, meditation, you name it. So this is good. So there is some agreement. There is also theoretical agreement. So most of the people who don't agree theoretically, they agree on the idea that complexity matters. This is a work by Matthew Larcombe. This is work by the group of, Global Workspace, Pierre Changeau, and this is a review that tries to find common denominators in the science of consciousness, and there's a strong convergence on complexity. Of course, there are different ways of measuring brain complexity. You could, for example, one way is to look at time series, EEG, fMRI, reconstruct connectivity graphs, and then use graph theory to compute complexity on the resulting graph. That's one way. Another way, a little bit more straightforward, is to look at time series and see how much information. That's, entropy, for example. But this is taking integration for granted. You don't know if all this information is generated by one system or a collection of many systems. And then there's a third way. So I'm now providing you with the rationale for TMS-EEG, the theoretical rationale. That is, you perturb the brain, you engage causal interactions in the network. So you make sure that what you're looking at is an integrated system of interacting elements. And then you measure the complexity of the pattern generated by this integrated system. So in this way, you capture unity and diversity at the same time. So this is obviously where TMS-EEG comes into play. And this is the level that I will describe now, where you perturb the brain, you extract this network causal response to the brain, and then you compress it to measure how rich it is. Again, unity and diversity. The rationale for this is that you perturb with TMS, you put your EEG cap, you record the deterministic response, your average response after many, many trials, and then you compress it, you zip it to derive an index that we call perturbational complexity index. If the system lacks integration, you have a local response in space and time and not much to compress. So the index will show you low levels, okay? High compressibility, low complexity. If the system is on the other side of the spectrum, one, but homogeneous, lacks differentiation, you will have a response to TMS that is global, but very stereotypical, simple, easy to describe because it's redundant, and again, low complexity. Only if the system is balanced between differentiation and integration, then the response is going to be widespread, and at the same time, differentiated. You will need a lot of information to compress it. And in this case, this PCI will be high. A couple of methodological remarks, because this is an hands-on session. Don't expect to put the coil on top of the head. Shoot at some suprathreshold intensity and then get an EEG response and compute PCI, because that's not the way it works. It's more like echography. You need to navigate. You need to make sure that you are hitting the brain right and that you're getting the right response as you measure, okay? So I'll give you an example. If you do this and you are online, and you will see how it works. You see online your response. You're not measuring much, okay? This is low signal to noise ratio. There is no response, although maybe you're stimulating suprathreshold, but you have the wrong angle, you are in the wrong spot, or you are not strong enough. You need to have an immediate readout of what you're doing. This is crucial. So if you are able to do so, like in echography, then maybe you can aim to a response of this kind. So you see a little bit more. But at the same time, you have a build-up of activity here, meaning that you are activating the brain, because to measure complexity, you need to first activate the brain, of course. So here there is some brain activation, but you also see that there is a sluggish, slow, central response. And Sara mentioned many artifacts. One of the most insidious artifacts is the sensory evoked artifact. TMS is clicking, TMS is touching. And you can have activations that are due to this on top of all the other artifacts. This is, for example, an auditory response. It's not TMS-EEG evoked. You won't measure complexity in this way. It's an auditory response. So there are ways to reducing this with noise masking. I'm sure you will see this. But also, you want to increase the strength of your cortical activation. So you play with the coil, you rotate, you increase noise masking, you optimize it, you increase maybe intensity a little bit, and then you are obtaining this. And this is a different story. So this is an input to the brain that is a good starting point for measuring complexity. It's not so difficult to get it. So rule of thumb that we use is that we want to have at least 10 microvolts peak to peak in the first 50 milliseconds. As a reviewer, if I see a paper where somebody is computing PCI and the peak-to-peak amplitude is 3 microvolt, I'm not sure you're stimulating the brain. Okay, so this is very important that you hit the brain well, that you have no artifacts, that you minimize artifacts, and then of course you need to measure the deterministic response of the network to the initial perturbation. So you need many trials. And collecting many trials, about 120 trials, takes time. So it takes 5, 6, 7 minutes. During that time, it is very important that the patient is in a stable state. If the patient is fluctuating between sleep and wake, or if he's not aroused, a patient is falling asleep, you need to arouse him and then go again. Okay, this is very important. Now, in practice, TMS-EEG, I'm sure you know, is made of a coil, an EEG system. You see it in the afternoon. A navigation system, very important to navigate around the lesion when you work on patient. And then an immediate online readout of your brain response. As I said, this is a crucial thing. Once you have it, and once you make sure it's good, then you can interpret it. And these waves, recurrent waves of activity, are speaking for interactions in the network. They are feed-forward feedback waves that are representing the echo of your brain, and you want to study and extract physical properties. And just to give you an idea, I'll show you this wonderful paper that recently came out from our collaborators in Boston, showing the connectome and at the same time the TMS-EEG activation source modeled. This is just to say you are exploring the connectome. Yes, the connectome must be there, but on top of that, you are really exploring the effective connectivity and the pattern of feed-forward and feedback interaction within this connectome. So it's a beautiful way of assessing brain properties. Most importantly, in this context, these TMS-EEG responses, they change dramatically when you fall asleep, for example. We're talking about detecting consciousness. First of all, you need to make sure that they work and they make sense in physiological situation where consciousness is there and it fades. So this is the example of sleep. So when you're awake, you have these recurrent waves of activity. You will see them today. They are differentiated, rich, widespread, high complexity. But then when the subject falls asleep, the connectome is still there. Nobody's cutting synapses, nobody's changing the connectome, but something changes in the postsynaptic properties of the brain. We'll go there, and everything is different. So you have this positive, negative, prominent wave. And if you look at the spatiotemporal dynamics by performing source modeling, in wakefulness, you have this interesting pattern of network activation. So you have this activation under the coil, then it spreads around. This is a deterministic response, very stable, which lasts for about 300 milliseconds. And it speaks for this recurrence activity in a complex network. And if you perform source modeling on the sleep response, it's a whole different situation where you have a strong local activation with the center mass under the coil that does not propagate. It stays there with a local maximum under stimulation. There's a major breakdown of effective connectivity in the thalamocortical system. This is true for sleep, but it's true also when you contrast different conditions where consciousness is there and not there. This is our benchmark. We are still in the validation phase, what I'm showing to you. So we tested the TMS-EEG evoked potentials in several conditions, not only wakefulness, but also dreaming. So you are temporarily unresponsive, behaviorally sort of paralyzed and disconnected, but you're dreaming. When you dream, TMS-EEG responses tend to be complex. Of course, I'm talking long dreams here. A short dream would be drowned into the 7 minutes of the recordings. But if you're dreaming, you know, that's what you see. Patients who are conscious in spite of brain injury, locked in, stroke, emergence from the minimally conscious state, so severely impaired but conscious, they also have complex response. It's an important validation because, you know, in spite of lesions, if consciousness is there, you have complex responses. And also then extreme dreaming in ketamine. Ketamine is a situation where you have dissociative anesthesia. So the patient is there, his eyes open. He can have surgery because he's completely disconnected from the external environment, completely unresponsive, but he's not unconscious. He's dreaming like crazy. He's just in another dimension. And also in this, you know, extreme case, complexity is high, which is a good, you know, validation. Then we have the counterfactual of situations where you are sleeping in deep non-REM sleep, anesthetized with different anesthetics, and you see this simple positive negative wave associated to low complexity. Of course, we can be very sure that subjects are conscious here. This is a methodological, almost philosophical issue. Can we be sure that consciousness is completely gone in these situations where consciousness is difficult to deny? It's weird, but it's difficult to deny because you're sleeping, your subject wakes up and says, no, I wasn't there, I don't remember. But maybe he had a dream, but he forgot, or a very short dream. So we cannot be absolutely sure that there was no consciousness. But it's the best counterfactual we can have. And we all know that in some phases, we are not there. So there is debate whether we are really unconscious in sleep and anesthesia. I think it is good to put forward this problem because we often dream, and that's been underestimated for years. And this is the reason why we took ketamine and REM sleep and, as a model. But also, we need to be aware that sometimes we are not aware, really. So when we sleep, non-REM, stage 3, this is a recent paper that came out. It's the biggest survey of EEG and dreaming. 70% of the time in deep non-REM sleep, people deny being conscious. Okay? Did they forget? We don't know. It doesn't matter. That's the best thing we have. So, very boring, but the validation is important. Once you have a validation, you use this benchmark to assess your normative values of complexity. So the red distribution would be the one you have in wakefulness, dreaming, all the conscious cases. And then this is the distribution of unconscious. Then you draw a cutoff. Operational cutoff is not the boundary between conscious and unconscious. It's a level that gives you allows you to do some inference. And if your patient is completely unconscious, it fails at level 1, level 2, level 3, but then you see the patient here, no? Knowing that this level of complexity are present only and only when you have some level of consciousness, then you need to question your clinical diagnosis. This is the principle. So, first of all, we tested for sensitivity in minimally conscious patients. So we took those patients who are fluctuating signs of consciousness. They're very difficult to detect, and especially those patients in which fMRI has a very low sensitivity, the P3B is often absent. And we asked, what is the performance of TMS-EEG in terms of sensitivity? And here, the performance is pretty high. This is around 94%, which is good. So it gives you confidence that you are not missing out many patients at this level. And then this is interesting because, as I said, sometimes you have a bad EEG in these patients, but in spite of a bad EEG, like slow waves, maybe at the surface of the EEG, you extract complexity with TMS-EEG. And this is important because TMS-EEG is extracting a deterministic pattern of activity above and beyond ongoing activity. So you can average out ongoing delta, for example, and if it's there, extract a pattern that is complex, if there is a residual complex pattern. So there can be dissociations between spontaneous EEG and TMS-EEG, and that's why there is an added value in perturbing on top of recording. It can also detect complexity when you are stimulating in minimally conscious patient islands that are pretty small. You know, sometimes lesions can be massive, and if you use the navigator and you perturb what is left, maybe less than one hemisphere, we know that one hemisphere is enough to support consciousness, you can still have high complexity. So it's a good assessment, provided you have navigation, of course. Now, I'll show you a couple of examples when we move now to the real thing. That is the inference we make in vegetative state, otherwise called, you see here, UWS stands for unresponsive wakefulness syndrome. That's the new name for the vegetative state because they're eyes open, but they are unresponsive. And it's a syndrome because we don't know whether they're conscious or not. This reflects, you know, the fact that we need to make an inference beyond this description. And the kind of inference we can make with TMS-EEG is quite interesting. So, for example, most of the patient, like 80%, they have low complexity responses. These responses are very similar to those that we collect in non-REM sleep or anesthesia. And I'll show you an example, and I will ask you to look at the screen at some point at the end of the movie and compare what you see on the screen to these two benchmark responses. So this is a patient with eyes open, unresponsive. Mario Rosanova is holding the coil. Navigation on an area of cortex is intact. We stay away from lesions, of course. We want a good entry port to the brain. And then soon you'll see what happens on the screen. Okay. Now, I think you can see this. Compare this one to these two. And what is the best match? Sleep or wake? I guess you see a positive negative wave here, do you? Which is very similar to this one. So this would be a confirmation of the previous levels, okay? But also, well, first of all, you know, I remind you, these patients are eyes open. It's paradoxical. It's a sleep-like response into an awake subject. We'll talk more about this. Now I'll show you another example. So situation is very different. So we have responses that are complex. They score above the threshold. And if you look at them, even without quantifying complexity, you see there are multiple waves of activity and so on. So the patient I'm going to show you, it's an extreme case among the many, where the patient is in the very acute stage of injury, eyes open. He was in the neurointensive care where the assessment was very unclear, inconclusive, and intensivists, they needed more information. So this happened in Milan, so there was a chance to move the patient to our center, intubated with, you know, intensivists on site, and perform the measurement to make an inference beyond what was known in the intensive care unit. And again, I will ask you to look at the screen. This is Gabriel, I think, holding the coil. And this is the response. Now, if you compare this to these two, you see there are multiple waves, different at different places. It's clearly this kind of response. So this is I just want to stress the point that you see things on the screen. You don't need to do statistics. Then you do. It's good to quantify, but you see things. This is electrophysiology at its, at its best. You see things. And what you see is informative. And in fact, this patient, after 3 weeks, recovered vertical eye movements and was able to communicate, and he communicated he wanted to undergo rehabilitation, for example. So the earlier you perform the measurement, the better it is, in principle. So there are reasons to make efforts to bring this in the intensive care unit, where the seizures are critical, and also withdrawal care is an option. More in general, TMS-EEG can enter the workflow of patients. So this is a comprehensive review of the electrophysiological techniques that are available to assess patients, and TMS-EEG plays a role, especially in those cases where the EEG is inconclusive. And I give you one more case. So basically, the idea is that you have a patient who's unresponsive, failed at all previous level. You open the black box of the brain by stimulating, and then you have the case of high complexity, low complexity. Cases of high complexity are cases where you need to assume that the thalamocortical system is okay, is in working order. The patient is probably conscious to some extent, but unable to show. And then you have to solve problems that are mainly problems with executive functions and communication channels. That would be the idea. Okay. And just to give an example, show you this case that is quite comprehensive. This was a patient that came in with a major frontal lesion due to rupture of an aneurysm. The patient was vegetative, also in the acute rehabilitation setting, so after weeks, vegetative. The EEG was severely abnormal. I'm sure you can appreciate a lot of slow waves there. The P3 was not there. So everything was negative. But TMS-EEG was positive. Twice in a row. Complexity was high. So this patient underwent a treatment with amantadine, which is pushing executive circuits. It's a dopaminergic drug, assuming that the problem was more with executive functions. And then it was scanned with fMRI. This was a luxury because it's not always easy to do fMRI in this patient. So the patient could go into the fMRI without sedation and perform the active paradigm as a confirmatory test that he was actually there. And he was asked to imagine to squeeze his right hand, and he produced a beautiful right motor cortex activation, confirming he was there. And he recovered then executive function, and then he recovered to a state that was an MCS plus state. So it's a state where you have response to commands. Then whether he could improve more, whether this is significant, whether the family accept this is a good outcome, it's a different story. Our job is to make sure that we don't let consciousness undetected. Then, the choice is up to family and caregivers, of course. But it's, reducing uncertainty, and it's useful. So what I've been talking so far is application of TMS-EEG in these extreme cases, disorders of consciousness here. But also, TMS-EEG can be useful after, when you are emerging, recovering consciousness, but you are still in a state where you have severe cognitive impairment, confusional state. One example is this paper by Bai that uses the same approach, TMS-EEG, assessment of the oscillations triggered by TMS-EEG, as well as assessments of complexity, to stratify patients for a risk of confusional state post-stroke. So there is also room for applying TMS-EEG in the assessment of throughout all the stratification of patients suffering from brain injury. And I'm slowly getting also to stroke, okay? This is confusional state. It's a major global impairment, often happening after recovery of consciousness. So this is another example. So we can explore the full spectrum of brain injury. But now I want to go to, you know, more into the mechanism of these observations. So this is the more diagnostic part. But TMS-EEG can tell us a lot about what's going on in the circuits. And indeed, what is very striking to us is the fact that these brains of people who are eyes open, the brain is intact, in portions of the brain are intact, but they react in a sleep-like fashion. So why is it? Can it be reversible? What are the mechanisms? Which leads me to the second part of my talk. Silvia? Should I finish? Okay. No problem. No, Madalena, don't worry. I just want to make sure. I have to finish before 3, right? Okay, cool. Now, these patients are sometimes, you know, they retain intact portion of the brain, metabolically active, but they're stuck in this sleep-like response, similar to sleep, midazolam, propofol, anesthesia. Others recover. This is work by Mario Rosanova. We don't know why. So what is it? So what are neurons doing here? And it's good to ask and go back to sleep, because after all, the mystery is in sleep. The connectome is there, as I said, but everything changes. So what is it that changes? So first of all, when we look at the physiology of TMS-EEG potential in very general terms, there are things that we have understood. So first of all, you fall asleep gradually. And when you fall asleep, your reaction to TMS is going from this complex set of recurring waves to a gradually simpler activation, which is initially stronger, then you have dampening of subsequent waves, to this extreme positive negative deflection. This is what happens as we fall asleep. When we are awake, we know, and we sort of understood by using modeling and other models, including animal models, that these recurrent waves are due to both cortical feedback and feed-forward interaction, as well as cortico-thalamocortical interactions. We know this because we performed a multi-scale work, TMS-EEG, intracranial in human and rodents, to assess cortico-thalamocortical. And there is a later wave here that is speaking for thalamocortical rebound. So there's a lot of stuff, both feed-forward and feedback. So it's an interesting assessment of thalamocortical system. Also, what we've understood is that when you fall asleep, the system goes from a feed-forward feedback model, a complex model where you have networks where you have both feed-forward and feedback reentrant interactions, building up complex patterns, to a feed-forward system. And we need to understand why. Why is it happening? So over the years, we've been developing an idea of what's happening. The idea is that the mechanism responsible for this collapse of complexity in sleep, connectome being equal, is the fact that cortical neurons, when you fall asleep, they develop some internal mechanisms that are called activity-dependent or adaptation mechanism. This is a classic in sleep neurophysiology, it's well known, by which they become active, or maybe they're activated, but they cannot sustain activation, and then they soon plunge down in a silent period. This is due to activity-dependent potassium currents, hyperpolarization current that becomes stronger if you have stronger activation, or more prominent inhibition. And these two changes are enforced as we fall asleep by changes in neuromodulation of noradrenergic, cholinergic, histaminergic, glutamatergic systems. So when they go down, you have increased adaptation. So the neurons are active and then they fall down into this period of silence that lasts for about 300 milliseconds. And this can be a killer for causal interactions because you have initial activation, neurons are receiving, and then they fall down, and then the feedback is impaired. So the idea is that this plays a major role. It's interesting because we know the cellular mechanism. It's interesting for sleep, potentially for brain injury. So what could happen, according to simulations of TMS-EEG in thalamocortical systems, is that when you're awake, you have, an initial input to the system, that is TMS, and then activity percolating through all the different cortical layers and cortical areas, and then the thalamus is involved, and you have this complex pattern. That's what we see pretty much in wakefulness. When you enforce these changes in neurons, postsynaptic changes, potassium currents, or increased inhibition, you have the initial activation and then a silent period, which definitely is a major blockage for building up long-range complex interaction. And you can probe this effectively with TMS. So this was modeling. In humans, can we detect the occurrence of these off periods? And off periods are interesting because they are, we think, responsible for the changes that we see in the response to TMS-EEG. But they're also responsible for delta waves when we fall asleep. Every time you have a delta wave, you have a silent period. So they've been studied in humans. And in humans, we have understood that you can detect them very easily by looking at the broadband EEG, so a prominent negative peak associated with a suppression of high frequency activity in the narrowband EEG. So the negative wave speaks for hyperpolarization. The reduction of high frequency speaks for reduction of firing. You can detect it in the EEG. So whenever you see a prominent slow wave associated with the suppression of firing, you can infer an off period. You can do this with time frequency decomposition. So this is the frequency, this is time, and you see that above 20 Hertz, at some point you have a prominent suppression of high frequencies, and then again resumption of activity corresponding to an occurring slow wave that is spontaneous and you can detect. But assessing the tendency of neurons and circuits to go into these down states that are killing complexity can be done better with perturbations. And again, TMS-EEG becomes very important here because I remind you, these are activity-dependent mechanisms. So they're better revealed by challenging the circuit and asking neurons to do something. They will become active and promptly go into a silent period if bistability is there. Indeed, we can trigger slow waves and down states even when they're not there. For example, in sleep stage 2 with TMS, this can be done easily, revealing bistability beyond spontaneous activity, precisely because of this activity-dependent mechanism. And there are many regimes where the brain is working, where you have a level of adaptation such that you don't see it spontaneously, but if you perturb, you reveal this tendency of neurons that then is detrimental for circuits. So it's good to detect it. So with TMS-EEG, you can detect this key neuronal mechanism, the tendency to go down, because you probe them, challenge them, and then you look what happens after. And at the same time, as we know, you can detect networks. So you are in a perfect position to assess the culprit, the neuronal mechanism, and its effect on the networks. And that's what I'm going to show you soon. So in general, during the Human Brain Project, which was a long project that lasted 10 years, we were able to test the idea that network complexity collapse because of these silent periods. So we replicated the TMS-EEG experiments at so many levels, intracranial, in humans, in animals, in cortical slices, in silico, replicating this change in the spatial-temporal dynamics, and then being closer to neurons, we could detect, the underlying mechanism, and this is what happens when you have collapse of network complexity. You have an initial activation, a silent period. The silent period is killing causal interactions. They are measured by the phase locking factor. I'm sure somebody will talk to you about this. It's a measure of causality that you can extract from TMS-EEG responses. And then in turn, if you kill causal interactions, you are preventing the buildup of complex patterns in the cortex, which you can quantify with PCI. So just to give you a graphical representation of what happens is that in wakefulness, very general, what happens is that neurons are sustaining firing, they sustain causal interactions, they receive the input by TMS, they propagate the input feed forward, and they also sustain feedback, building up complex pattern of reentrant activity. And this corresponds to a complex system. If you fall asleep, the simple fact that you have increased potassium currents in neurons or increased inhibition results in a downstate after the initial activation. You have the feed-forward sweep, yes, we see an activation, no doubts, but then the feedback is coming back into a refractory period and it dies. So the networks turn from feed-forward feedback to purely feed-forward. There is one thing we all agree in the field of consciousness: feedback, reentrant activity is key for consciousness. That's the only thing we all agree. So En-TMS-EEG provides you with the perfect measure of feed-forward and feedback, because there's nothing better than perturbing and looking at what comes back after the perturbation. I'll skip this. Beautiful work by Andrea Pigorini, who was responsible of guiding this human brain project, largely responsible for guiding this human brain project exploration. Just to move to patient, to show you how this is relevant, for example, in the vegetative state. Vegetative state, patients are, if you compare them to somebody who's asleep or awake, they look as if they are asleep. By all means, they have a slow response, positive, negative, suppression of firing. Although their eyes open, I remind you this. And patients who are recovering from the vegetative state, they start with this low complexity response, suppression of firing, short causal interaction, low complexity, and they gradually get rid of this neuronal downstate to recover. So it's reversible. There are two messages here. One, sleep-like cortical mechanism can intrude into the awake brain of somebody whose eyes open. Two, this is a dynamic process that is potentially reversible. Vegetative state, everything is in the same situation. So wherever you stimulate the cortex, you have this sleep-like response. It's a global phenomenon. But then more recently, and this is an important paper by Simone, we understood that these sleep-like responses can also be local following focal injury. So this is generalizing the concept of an intrusion of sleep-like activity to stroke and generalizing the applications of TMS-EEG. So whatever I said before applies also to stroke. You see that in the perilesional area, you have, again, this slow wave response, and you also have low local network complexity. This is detected even beyond spontaneous EEG. So even in cases where you don't have spontaneous low wave in the EEG in the perilesional area, you reveal them. So you see here, the perilesional area is clearly showing a slow wave, whereas the spontaneous EEG does not show. So you reveal by stability beyond observation. But most importantly, these alterations, and actually the reduction of this alteration, correlates with recovery. So patients who are recovering over time, as well as, similar to what I've shown in the vegetative state, but now only local in the perilesional area, they get rid of the downstate and their complexity increase. And this reduction of slow waves and increase in network complexity correlates with recovery. This is an important marker. Potentially, this is a new, very informative electrophysiological marker of the consequences of stroke, and also can guide us towards treatment. I want to just say that the TMS-EEG community has been struggling a little bit with reproducibility and methodological issues, but it's now growing. And I think these meetings are helping a lot, as well as the meetings that are taking place in Helsinki. And things are getting way better. What's happening, for example, we met recently together with experts of stroke in general, Ulf Ziemann, Friedem Hammel, Scherpel from Frankfurt, as well as Christian Grefka, Sylvain Arquel, and Giacomo Koch, many others. And we found out together that we are all converging on a similar marker of stroke changes in excitability. So we all see this positive, negative wave. Beautiful work from Germany in general. So this is a good news. I think, for the field in general. Of course, there are many open questions. So to finish, why should sleep-like dynamic intrude into wakefulness after injury? There are many ways. So one way is that you might have lesions to activating systems directly. That's a very trivial manner. Another way, you can have lesions in the white matter interrupting ascending fiber from the brainstem to the cortex. This is more subtle because you can have white matter lesion and part of the cortex falling asleep. You can have diffuse axonal injury, which often happens after traumatic brain injury. So ascending fibers, mainly increasing potassium currents. But then you can also have damage to lateral connectivity. So, for example, the perilesional area becomes disconnected because of the lesion and lacks excitatory input. An area that is far away but strongly connected to lesion becomes deprived from excitatory input and disfacilitated. And if you have less excitatory input, you have more inhibition. So your balance shift towards inhibition, which is also a factor promoting downstates, or you can have lateral axonal injury in traumatic brain injury. And indeed, every time the cortex is disconnected, you have this kind of bistable slow waves. We know this in cortical slices. If you cut the white matter under a gyrus, if you cut and isolate one entire hemisphere, this is work by Michele Colombo that we recently finalized in hemispherotomy patients, so where the one hemisphere for treatment reasons is isolated from the other hemisphere and from the subcortical structure. It's not silent. It's going into these up and down states between activity and silence because of adaptation. So this kind of mechanisms are enforced every time you have some level of differentiation. And again, TMS-EEG and neuronavigation, how important this is in stroke. Because apart from these, extreme situations, if you have, imagine that you have, actually, not imagine, this is a real case. You have a lesion in the white matter, a small lesion, and then you can reconstruct to tractography, which is the cortical area that gets deprived from ascending fibers the most. You put it on the surface, and then you load it in the navigator, and your inference would be that the prediction would be that this area is asleep. Although the subject is awake, the cortex is intact. It's just deprived from activated system, whereas these two control areas should be awake. So if you do TMS-EEG here, in one area that is not disconnected, you see wake-like response. Subject is conscious, is impaired, but is conscious. You see a wake-like response, maybe just a little bit slower on the contralateral side, but then if you shoot on the disconnected area, you see a sleep-like response. And this is very revealing of what might happen after brain injury. It's not only the injury, but it's the consequence of the injury far away that is falling asleep on some part of the brain. And this might have an impact on behavior. This is work by Caroline Sherper, beautiful work showing basically the same thing. Strokes disconnecting cortical areas are leading to sleep-like responses in the deafferentated area. There is also inflammation that can play a role in promoting this dynamics by increasing potassium channels as well as hypoxia. So there are so many ways after injury that can converge into promoting sleep-like dynamic intrusion in the awake brain with all the consequences, and we can assess this with TMS-EEG. Now, just to finish and try to what do we do with this? What are the final remarks about, in general, this idea revealed by TMS-EEG? So first of all, the general idea would be that you have an injury, you have differentiation from ascending fibers, lateral differentiation, maybe inflammation, hypoxia. For many reasons, you have a donut of the brain around the injury that is falling asleep. By falling asleep, I mean not real sleep. I mean cortical sleep, the tendency of the neuron to go into a downstate and react with a slow wave and a breakdown of interaction. That's what I mean. This is local, but it can also act like an epileptic focus. And we know that slow waves, once they are generated somewhere, they can propagate and interfere. They do so normally when we fall asleep. So slow waves are generated here, they propagate there. here and they propagate here. They do this every night in our brains. They travel. Can they also travel and disrupt and interfere with networks far away after a lesion during wakefulness? So this question was addressed thanks to, again, Simone Russo, Andrea Pigorini, who's here. Simone is now in the US, unfortunately, who were able to study a very specific intervention that is called, and Ezekiel, who's also here, I think. They studied a very specific kind of manipulation that is called radiofrequency thermocoagulation. It's a small lesion that's performed in humans to treat epilepsy, but it can also be a good way of studying the consequences of brain injury, because you have a baseline, you have an after, and in the after, slow waves are strongly developing around the injury, 1.5 centimeter around the lesion, you have slow waves, but also they were able to demonstrate that they propagate far, and they do propagate to sites that are connected to the perilesional area, that is the focus of generation, because they propagated where the cortical evopotential was a positive connection. So this is I think, offers a good explanation of the network effects of brain injury that are functional and beyond structural injury. There is also reason to think that slow waves, once they're generated, by stable behavior neurons, can be very detrimental for behavior. We know this not from stroke studies, because they haven't been performed yet. They should, they will. But we know this from studies in sleep deprivation models. So if you sleep deprive a rat, and then the rat has a slow wave and a silent neuronal period in the left motor cortex, in that very moment is unable to reach a palate. So there is a impact that is topographical specific and time specific. Downstate, neuronal silent period, no reaching. The same occurs in human in the cognitive domain, frontal slow waves, parietal slow wave with different cognitive effects. And it's been studied also intracranially by Yuval Nir. So can you imagine what slow waves massively intruding after brain injury can do to behavior and to symptoms? That's never been considered, but it should. For example, in neglect. And we are starting to do this for now, looking at spontaneous slow waves. For example, this is work by Elisabetta Litterio and Elena Focacci and Maddalena Bucur, where in neglect patient, we are studying the degree of neglect, so attentional bias. They are looking mostly at the right. That's a problem when they are in front of a starry night. And they have a lot of slow wave on the right side, by the way. So it's not only the injury, but there is, sleep intrusion into the right parietal frontal network, which the hypothesis is could partially explain part of the symptoms. And indeed, the preliminary evidence is that when patients are recovering and they look more to the left, they have less slow waves. And this is interesting. And it could be also probed with TMS-EEG even more effectively. Which leads to intervention. Speaking of TMS and TMS-EEG, one would want maybe to kill slow waves and to reduce them or block them. There are ways to do this in vitro. There are ways in humans to enhance slow waves. That's been done for the sake of improving sleep, but there are not many studies in reducing slow waves. We should do this and test whether this has an effect in patients and on recovery. But we should do this having in mind that things are more complicated than this. As I said, these are sleep-like dynamic. And sleep is bad for networks, even physiological sleep. When I fall asleep, my cognitive functions are dramatically impaired, I'm motorly impaired, but it's good for the brain. I wake up with a better brain. So sleep has a good side, actually, definitely has a good side. What about slow waves or this cellular dynamics after injury? Are they good like sleep also? Do they have a protective role? The question is open because slow waves and down states are associated with less metabolic demands. It can be useful after brain injury. They've been associated to waste management in the extracellular space, which might be good after injury. They are associated to homeostatic plasticity, which might be good after injury, and axonal sprouting, as well as a protective role against epilepsy. So, bottom line is TMS-EEG, with its direct assessment of cortical excitability, input-output properties, adaptation, tendency of neurons to become silent, and then connectivity. It's a powerful, powerful tool. It generalizes well beyond disorders of consciousness. It goes into the field of brain injury as a new tool with potential applications in understanding what happens after brain injury in general, in developing new markers that are diagnostic and prognostic, and especially in guiding treatment, something that we lack. And it's probably, this has been hampering the field of stroke neuromodulation for many years. So that's an opportunity. Many things to understand, evaluate the protective role of slow waves, whether we should kill them, maybe promote them during some phases, in the acute phase near the injury, where they're needed, but if they persist and if they go far, we need to block them. So it's all open. We're going to explore part of this in an ERC grant together with other collaborators. Maurizio Corbetta is a neurologist, expert in networks. Mavi Sanchez-Vives, she's an expert in the cellular physiology of sleep. Gustavo Deco is a modeler. We'll do our best. But I just want to remember that these ideas, whatever I said, apart from TMS-EEG, is not new, because the concept of network impairment after brain injury, functional network disruption, was put forward in 1914 by von Monachov, sort of forgotten. The fact that slow waves are intruding into the brain after injury was observed in 1934 by William Gray Walter. And if you put it together, you have network impairment, sleep intrusion, and we know what sleep does to network. It's easy, but it's easier if we consider the discovery by Mr. Asteriade, who discovered the mechanism, the cellular mechanism of slow waves. Slow wave is a silent period, and the silent period breaks the network. And finally, I think the important piece in this context is that we can explore all of this network, bistability, cellular properties with TMS-EEG. So good luck. Enjoy the hands-on session.
Okay. Good afternoon. So the afternoon is mostly dedicated to the live demo session, where you will see live what we do every day in our TMS-EEG lab and what is our approach to record reliable EEG responses to TMS, TMS evoked potentials, TEPs. While colleagues of mine, Silvia, Gabriel, Giulia, Letizia, and many others will check whether they prepared everything for the live session during the Marcello's talk. They will check now for whether the impedances are acceptable, the signal is good. They will then co-register the head of the subject with the neuronavigation. Meanwhile, I would like to provide you with a short introduction to TMS-EEG. You had already some hints by Marcello that was showing how to use it to study the electrophysiology of brain lesions, such as stroke or lesions, more severe lesions that bring to consciousness disorders. Before starting, let me disclose that I'm also scientific advisor and shareholder of Intrinsic Powers, which is a spin-off of the University of Milan. Now, this is the outline of my talk. And let's start by describing the techniques. So you already heard about TMS-EEG, which comes from the combination of TMS, transcranial magnetic stimulation, that is exploiting the Faraday's principle of electromagnetic induction. Henri Hannula described that very well and extensively today morning. So it uses time-changing, high-intensity electrical current that is moving within two copper coils, and then this is inducing a magnetic field that in turn induces an electric field, and this third one that activates cortical neurons under the coil. And actually this year we are celebrating 40 years from the first publication ever made by Barker, Freeston, and Jalinus, showing that by stimulated with a circular coil, which is different from the one we are going to use today. This is a figure-of-8 coil. It's more focal rather than the circular coil that stimulate a large area of the cortex. Here you have Timothy Barker stimulating a healthy subject and recording from the periphery the motor evoked potential produced by the activation of the cortical spinal tract. We know now that TMS is able to activate cortical neurons. We know that from animal studies in cats, in non-human primates, and rodents. And the very first action potentials that follow the TMS discharge are already occurring after 2-3 milliseconds and can last up to 100 milliseconds. And we have today beautiful models at the single cell level where you can see, let's see if it's going to work, this one. Yes, you have time here. At 0, you have the TMS pulse. The axon is the first structure activated by the TMS coil, and then you have the electrical signal invading the neuronal body. This is a pyramidal neuron and all the dendritic branches. On the other side, you have the EEG, and the EEG is actually recording the postsynaptic potentials produced by pyramidal neurons that generate, because of their geometry, that generate open source electric fields that can be then, since they are open electric fields, can be recorded from the scalp. And this is a very personal hall of fame of the great neurophysiologists that preceded all of us, Hans Berger, Edgar Adrian, Alfred Loomis, and Walter Gray Walter, that first recorded EEG in humans after Caton did that in animals, and showed that the brain is able to generate alpha waves, spindles, and delta waves, again over a lesion in an awake patient. This was the seminal work by Walter Gray Walter. This is another beautiful paper coming from the 60s, where Otto Creutzfeldt was able to record intracellularly the activity of pyramidal neurons and the EEG on top of them, of those neurons, showing that the EEG is coming from the synaptic activity, and is a reflection of EPSPs and IPSPs. Now, this is the first paper combining the two, the TMS and the EEG. is a very old paper coming from the late 80s from a group led by Krakow, Amassian, and Maccabe, New York University, State University. They were able to stimulate one side of the brain and record the EEG responses from the other side of the brain. And I always like to start from this paper, first because it was the first trying to combine EEG and TMS, but second because if you go if you focus on the method section, there are some lines that show already what you are going to see in the live session and what is the general approach to record the reliable and reproducible EEG response to TMS. The method section reads, during delivery of stimuli, the magnetic coil, this was the way to indicate TMS at that time, was progressively tilted towards the horizontal until a response, an EEG response, was elicited, and then carefully rotated to yield the minimal stimulation artifact. Additional stimuli were then delivered at surrounding sites at approximately 1 centimeter intervals until an optimal response was recorded from contralateral homologous scalp. So this little paragraph from the method section of that paper is already showing that the operator was searching for the response. The response was not there, was not immediately there. You just put the coil and to record the response, but you have to look for that, okay? Of course, in that case, the response was recorded on the other side of the brain. But as Marcello showed before, now we are interested in measuring cortical excitability of cortical neurons under the coil, so a more direct measure of excitability. And we had to wait for this guy, a physicist, a Finnish one, from the University of Aalto, that devised the first fully TMS compatible EEG amplifier. This is the little circuit that was mentioned by Sara Pizzamiglio today morning. It's a sample and hold circuit, is out of production, but was able to, for the first time, provided us with immediate response or early responses, EEG responses to TMS, such as the ones you have here. So this is a TMS evoked potential recorded under the coil after a parietal cortex stimulation a few milliseconds after the TMS pulse. While, and this is also a picture you already seen today morning in the Sara's talk, while if you're going to use, let's say, a traditional EEG amplifier, the one you use in your clinical routine, you would have saturation of the amplifier. So the EEG is basically blind to the brain response to TMS for 5, 6, 7 seconds. In the last year, inspired by one of the first prototypes by NextTeam, we actually devised, and this is a paper by Silvia, we actually devised a graphic user interface that is allowing you to guide the process of searching the response that you just saw in the method section in the Krakow's paper, looking for the response in real time, as they were doing now more than 50 years ago, 40 years ago. So you can look for a response, and in that case, you want to be sure that you are effective on the cortex, you are producing some responses. You will see now this in action in a few minutes, and that you were good in controlling or abolishing confounders such as auditory evoked potential that come along with the TMS coil discharge. And here again you have one of our PhD students, Julia, that will take part into the live demo session with the structural neuronavigation. Here you will see exactly that. And the functional neuronavigation, if you want, where you can see already in real time, only after average references as the only one preprocessing step there, you can see the TMS evoked potential. Here we were targeting the premotor cortex. You will see all these steps in a few minutes. But if you want, you can go also through this tutorial performed by Silvia, I think a couple of years ago. The idea is to have always a rough but reliable idea of the quality of your signals. So in real time, thanks to the graphic user interface that is now embedded into the devised by Silvia, but it is now embedded into the nTMS system, you can have an immediate idea about the effectiveness of your stimulation and absence of confounders, such as TMS-related auditory evoked potentials. And you can see in real time, Marcello showed some examples of that, of this. You can have in real time an idea of whether the patient is maybe is producing complex waves like those ones, or simple slow waves, such similar to the ones that you can record in most of vegetative state patients. And now this graphic user interface was implemented also in a more automatic way by the group led by Risto Moniemi. And it was very recently used also to record EEG responses to TMS from the dorsolateral prefrontal cortex, which is, maybe we have time to show that, which is kind of a tricky target, but very important for research in research that are for researchers that are trying to look for, that are looking for treatment for major depression, for instance, in the psychiatry field. Now, what measures you can extract from the TEPs? So you have here the same TEPs I've shown before. You can measure the latencies here of early and late components. Usually early components are occurring within the first 50 milliseconds, 50, 80 milliseconds, while late components are from 80 milliseconds up to 300, 400 milliseconds. You have oscillations. You have immediate components that occur like 2-6 milliseconds after the TMS pulse. And for instance, you can measure the peak-to-peak amplitude, and this will be one of the measures that give you an idea of cortical excitability. Or you can measure the slope of these very early components. You can measure, you can see whether there are differences in the waveform by stimulating different cortical targets. And generally speaking, we know that the more posterior you are, the slower responses you get after the TMS pulse. Or you can look at all channels and, oh, sorry, this is a measure that captures this slowing, these slow responses over the more posterior cortices and faster over the more anterior ones. But you can look also at the all channels and check whether, just a second here, and check whether these waveforms are corresponding to different topographies, looking at the whole scalp. And actually what we have observed is that if you have a premotor, motor, parietal, and occipital target, you have slower response here, faster here, but the topography is, these are single subject, sorry, responses recorded at the single subject level. You have topographies that are changing, that are different, comparing different targets. This idea of the differentiation of the EEG response to TMS was challenged, say, around 2018, and this group of researchers were suggesting that actually you do not have this kind of differentiation. So we worked on that, and we came up with an experimental protocol, a kind of complex one, where we were systematically comparing responses, EEG responses to TMS obtained by targeting a premotor and a motor cortex, and then comparing responses to electrical stimulation, realistic sham TMS, again from the two target, and eventually from high intensity stimulation of the scalp. And we came up with results. This paper is under revision now. We came up with these results. When you compare the EEG responses to TMS from the premotor and motor cortex, you have significant widespread spatiotemporal differences up to 300 milliseconds at both group and single subject level, while you are stimulated with the realistic sham TMS and high intensity scalp TMS, you have no significant or marginal differences, provided that confounders and artifacts are properly and effectively controlled for. We'll come to this point in a while during the live demo session. And these are the results, some of the results of the paper, where we exploited a tool that is also reported in this paper, that is under review as well by Bruno and other colleagues of ours at Aalborg University. Bruno is around, is there. Okay, shake your hand, Bruno, for the audience. Okay, you can ask questions to Bruno about this beautiful tool that is able to extract the most reproducible components of TMS-evoked potentials across trials and subjects. And here you can see that by stimulating the premotor cortex, you have this component that is prevailing over the others. Is the most reproducing. It's not prevailing, but it's most reproducible across trials and subjects with this waveform, this main topography, and these components, which is completely different for the first one when you stimulate the primary motor cortex. Then you can be interested in extracting other indices, for instance, connectivity indices, but of course you have to go down to the cortical sources because of the volume conduction effect that can be affecting EEG signals. And you can come up with these nice maps. This figure coming from an old paper by our lab, 2009. And these activations of the cortex are probably due to reverberating responses of the brain to the initial TMS pulse, and as has been shown in this modeling study or this study by the colleagues of ours at Iowa University combining TMS and intracranial recordings. And actually from those maps, you can come up with the synthetic indices of connectivity that were published in 2010 by Adenauer Casali. And you have these SCD, BPL, and SCS, which are indices of excitability and connectivity at the cortical source level. The very last part of my talk is about a series of applications. So in 2019, this consensus paper came out on clinical neurophysiology, reporting a series of application of TMS-EEG in neurophysiology and clinical neurophysiology. For instance, you can be interested in measuring cortical excitability across the sleep-wake cycle, as we did in 2013 with colleague of our Reto Huber, or you can be interested in measuring changes in cortical excitability during and after a TDCS stimulation protocol, or you can be interested in measuring natural frequencies, meaning the main oscillatory component of TMS evoked potential in clinical populations such as schizophrenic patients. Here as shown, as has been shown by Fabio Ferrarelli and Simone Sarasso, who is there. Shake your hand, Simone. Thank you. They were nicely showing that this sort of gradient with slower responses in the back and faster in front was basically not there anymore in schizophrenic patients, and there was a nice correlation with some clinical indices. Or you can be interested, as the group of colleagues of our Aalborg University were doing, can be interested in measuring changes in cortical excitability while you are inducing some pain perceptions. You can talk to Enrico and Daniel. Shake your hands. Okay, thank you. About this beautiful paper. But there is a paper that I like even more. That is this one, that is suggesting that you can predict somehow. I hope Daniel, I guess Daniel is going to talk more extensively about that tomorrow, maybe. Okay, you can predict whether a patient is going to be a responder or not affected by chronic pain, is going to be a responder or non-responder based on the connectivity pattern that you can measure before the therapy. Or you can be interested in measuring cortical excitability while the patient is intaking a dopaminergic drug, as we did in a Parkinson's disease patient now in 2019. You may want to measure cortical excitability in Alzheimer's disease patient and the connectivity patterns that come out comes out after the treatment. Or you can be interested, but Marcello talked extensively about that, in changes in excitability and TMS evoked potential waveforms in wakefulness and sleep. So when the subject is changing his brain state. Marcello talked already about that. So you can be interested in measuring cortical responses in vegetative state patients, as we did now in 2012 and was confirmed by another paper in 2013. And you may want to measure complexity indices extracted from these TMS evoked potentials. And actually both This perturbational complexity index, again, devised by Adenauer-Casali, and the natural frequency was used to predict the occurrence of delirium in stroke patients. Very last slides. You may want to actually this approach, as Marcello was already showing, we use this approach in patients affected by severe brain injuries, like in this case. And now colleagues of ours at the Harvard University are applying this approach in the ICU. And then you may want to apply TMS-EEG in stroke patients and measure excitability and connectivity changes in stroke patients, as I think, Simona, you are going to talk about that in the next days. Okay. And this is just an overview of the measures you can obtain from TMS-evoked potentials. These figures are part of the new version, the new release of the consensus paper by Tremblay. The first author will be Ulf Ziemann, the last author will be Risto Ilmoniemi. And let me thank, and I'm done, the beautiful group of people I belong to. And let's start with the live session. Thank you.
So we are almost ready to start with the hands-on session. Here you have several pieces of the whole equipment, because it's easy to say TMS-EEG, it's a short acronym, but actually it's made of several parts. So we have the EEG, so the proper amplifier is on this table with a battery pack. Then we have a computer which is connected to the amplifier that stores data on the hard drive. Then we have this unit which mixes both the TMS stimulator and the neuronavigation system. The first thing we have to do is to select on the MRI of the subject that's being loaded here 3 anatomical landmarks. These landmarks should be, on the head surface, because afterward we have to reach them with this wooden stick. You see these gray markers on this wooden stick? They are visible to that infrared camera. So we will have the subject wearing a small device, which is like this, which will be attached to his head. So the camera knows in the space where the subject's head is. And then we will touch the same anatomical landmarks on the subject's head, such that the neuronavigation software can match the real subject's head with the MRI head model. So at that point, we can know exactly where we are going to stimulate on his brain. So let me select these points. We usually set left and right tragus and nasion. So okay, left tragus is set. I go right. Set. And then the nasion. Okay. Now we can activate the tracker and put it on the subject's head. Can you hold it? field of view of the camera is correct. So we can get the position for the camera. Okay, sorry. A position for the camera such that the subject is located in the correct field of view. Okay. Now we use this stick and we touch the same spots on the subject's head. Mario, can you help me by just pressing the pedal? Okay, so I just touch the left nasion, the left tragus, sorry. Okay, you can click. Okay, yes. And then nation. Okay, you can go, Mario. And then the right tragus. Ok. Più verso così, ok. Okay. Now the software asks for 9 additional points on the subject's head. So now the navigation knows where the subject's head is, but if I touch those additional 9 points on the subject's scalp, the neuronavigation knows what's the shape and size of the subject's head, and it will use it to estimate the volt per meter, that is the intensity of the induced E field on the cortical surface. Of course, to save computation time, the neuronavigation in this case makes an approximation. So Gabriel's head is approximated to a sphere, concentric sphere, multi-layer. So it's a coarse approximation, but it's something that we can have in real time. That's the advantage. And then offline, you can make it more accurate. Grazie, Mario. So I just touched the first one, okay. The second, the third, okay. Fourth, it's enough that I center, okay, that I go in the darker areas. Okay. 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 If you have questions, please stop me at any time. Okay. So the mismatch between the 3 anatomical landmarks and the 3 landmarks taken on the subject's head in this case was 1.6 millimeters overall. So it's very good. It would not accept an error larger than, if I remember correctly, it's 3 or something millimeters. 2 millimeters, okay. Now, so far we have focused on the left screen, where you see the MRI of the subject in the classic 3-slice view, sagittal, coronal, and axial. But then here, I can enlarge it, you can see a 3D rendered view of Gabriel's head. So we can conveniently rotate it such that on the screen we see Gabriel and as we see him in reality. And we can also pretend to peel his skull and scalp, and we can expose his brain. Okay, so his frontal, occipital, left hemisphere, and right hemisphere. Now we can choose a cortical site that you want to stimulate and proceed with you know, coil positioning and all the rest. Simultaneously on the other screen, we have the EEG, which is continuously scrolling. Now it's, say, resting state EEG, but then as soon as we start delivering pulses, we can ask that software to plot the average response across different pulses. And we will show you how we'll do that in a moment. Okay. The software that you see on the right screen is a real-time graphic user interface that allows to visually inspect TMS-EEG data already during the experiment. And when you will see also the data analysis sessions tomorrow and on Wednesday, you will see that after the analysis, if we were able to control for artifacts in real time in an optimal way, you know, the waveform of average TEPs after preprocessing will be very similar to what we see today just on the screen. So that graphic user interface does not save anything, it's just for visualization. Raw data are recorded on this computer, which is directly connected to the EEG amplifier. Okay. Okay, now, this visualization mode is good for looking at raw EEG, but it's not optimal for looking at TMS evoked potentials, because in this case, we need to go and see average responses. Now, Mario, which site are we going to stimulate first? Well, the first the first application of TMS was stimulation of the primary motor cortex, right? So we could first find the primary motor cortex, the hand M1, so-called hand M1, and start stimulating and see the peripheral effect. Now, for the sake of time, you are not going to put electrodes on Gabriel's hand, so we will see the twitch. In this case, it will mean that we are stimulating above the resting motor threshold if we see the twitch. But we want to make sure that we are stimulating the primary motor cortex. So we'll do like this. Now Gabriel is listening to what we are doing, but unfortunately during the real experiment, he will not hear us very well because he will wear in-ear earphones playing a masking noise that's been specifically designed to mask the TMS click. In fact, when you start delivering TMS pulses, you will hear a loud click sound produced by the coil vibration within his cage. So this type of noise is a click, and of course this stimulus occurs time locked to the real TMS pulse, which is the main stimulation we want to deliver to the brain. As a consequence, the brain will respond to this auditory stimulation, producing an auditory evoked potential superimposed linearly, non-linearly, we don't know, to the genuine brain response to direct stimulation with TMS. So in order to get rid of this auditory evoked potential, we need to mask it by having the subject listening to this noise. It's possible to titrate the characteristics of this masking noise very carefully depending on your stimulation. We have developed a software tool, MATLAB-based software tool that can build a customized noise such that we can properly mask the TMS click without increasing too much the masking noise loudness. Today, you know, yeah, please. The noise is a mixture of white noise plus the click sound of the TMS coil replayed continuously with the phases shuffled. So you transform the noise waveform, the click waveform into the frequency domain, then you scramble phases and you replay it continuously overlapped with white noise. And you can also choose how much white noise, how much click noise you can mix to optimize masking. So we already did this job, and I can simply let you hear the noise click. Sorry, it stopped for some reason. So this is like, I don't know, waterfalls thing. Okay, Gabriel will listen to it. Otherwise, we would have this auditory evoked potential. So I think we can start with Mario, would you like to start with motor cortex, or would you like to show the difference between auditory evoked potential without masking and with noise masking? Okay. Is somebody interested in the presence of the auditory evoked potential? Yeah, please. Okay. So if we start stimulating without having Gabriel wearing the in-ear earphones, we will see after a few trials the presence of a slow and late component with a frontocentral distribution mainly.
And, I will use the coil in, say, in a sham position, meaning 90 degrees over the head, touching the head. This way we preserve the vibrations made by the coil and the bone conduction of the TMS-related click, but without stimulating the cortex.
So 20 pulses have been delivered. And what do you see on the screen? Not much, right? Make it.
And how zooming in with the camera from the room?
And what about if you put the phone here? Maybe some a bit better? No, eh? It's a resolution, yeah, aliasing, I guess. It's true. Ah, I see, I see. Well, you all may see that at least there are a lot of vertical bars on the screen, which basically prevent to see anything useful. Those vertical bars, guess what it is? Sorry? The ringing artifact, so the pulse artifact, which is ringing a bit, and it happens in all the channels because it's huge compared to the EEG response. So forget about that for a while. And now Mario zoomed over a single channel to show what.
So you can see, besides the step artifact, as the colleague of yours said, there are some deflections, kind of late deflections. Those are not coming after the TMS pulse, but they are they reflect the auditory evoked potential. We know already that the topography of the auditory evoked potential is very central, so is involving central electrodes at the vertex. So that's why I zoomed on those electrodes, on that electrodes, but those are not responses to TMS. They are responses to the TMS-related click. So now what we are going to do next is first to remove just graphically the step artifact, and then to play noise masking that is meant to abolish the brain response to the TMS click. This is let me note that let me remark that the removal of the step artifact is just graphical. Everything is going to be recorded. Okay, that is only to show the very first milliseconds after the pulse, and this will be very important for two reasons. First, to see whether there are scalp muscles. It's not the case here because we use the coil in a sham position, so nothing is going to be excited under the coil. And second, to see whether there are responses to the TMS.
And so in this display, positive is upward, negative is downward, and this component is around 160, 180 milliseconds. So it could be the P200 auditory component. Now I just clear the average, and I think, yes, Gabriel, is noise on? No? Now? Is it on? Wait. Noise masking. Try another cable. You mean the X10 cable? Or we will take the other cable. Yes, this is okay. Thanks. Qui. Ok. Sai cosa? Now it works. Okay, so maybe also with the extension cable. So it was just the computer that was not like that. Okay, so now noise masking is on, so we can try the same. So again, sham stimulation, but with the noise masking on. Do you remember that component? We can see whether it's still there or not, whether noise masking is effective. You see the subject is complaining because he says that he's hearing the click. In this case, we have a collaborative subject, so we can trust him and we can decide to increase, in this case, noise loudness. Wait a minute, Mario, just clear the average and we can increase a bit noise loudness. More. Okay. So we can retry. We can average the same amount of trials we did before, so 20 trials. We are delivering TMS pulses about every 2 seconds with a slight jitter. If we zoom on the same channel that we were highlighting before, you see that it's much flatter than it was. So in this case, we have a collaborative subject we could have asked, but if you don't have a collaborative subject, it's good to test and try in this way. It's very fast. It took more than needed for the earphone problems, but it's very fast. Please. Well, now we cannot superimpose the two, but it could be a nice feature, you know, for the graphic user interface to keep in memory something that you like or you want to keep and overlap it with the next one, just as a comparison. I think it would be a very nice add-on, very useful, yeah. So today you have just, you know, to remember yourself, but it would be good, yeah, for sure. Okay, so now that we know that noise masking is effective, we can start stimulation. You see that in this case, I have increased the stimulation intensity at 100% of the maximal stimulator output. This, of course, maximizes the loudness of the TMS click. So in this, since I think we are not going to stimulate at this high intensity in this subject today, we will stimulate lower, so we are pretty sure noise masking will be effective.
So now we are going to target the motor cortex, which is a traditional targeted field, and this will help us in showing some of the troubles issues you can encounter during TMS-EEG measurement. Okay, so
we can locate on the cortex the hand area, and we can start stimulating at, say, a mid intensity and see whether we can get a response. So this is, let me see, better, yeah. This is likely the central sulcus. So we know the primary motor cortex is right forward, frontal to the central sulcus. We can find it. And you see that in real time, the neuronavigation software displays where we are stimulating. As Mario moves the coil on Gabriel's head, also the colored map of the induced field moves. Is it moving? It's moving. Okay.
So from the subject behavior, we have the proof that we are over the primary motor. Sorry, from the subject, based on the subject behavior, we have the proof that we are stimulating the primary motor cortex. He was Gabriel was signaling that was reporting that the thumb was twitching, so we are okay. We may want to lower the intensity of stimulation to avoid the peripheral twitch that is also associated to somatosensory evoked potential, because you have the activation of neuromuscle spindles and the reentrance of somatosensory input from there. So you will have again TMS evoked potential plus something else, something coming from the periphery. So we will lower a bit the intensity, but what is most important now is to show what we see from the graphic user interface in real time.
You mean?
Now, we are going to comment on what you see on the graphic user interface. The scenario is completely different. The picture is completely different. You have some very large spikes over the left part of the scalp, there where Silvia is moving the arrow, the pointer. Okay. Considering that we have visually removed the step artifact, what are those spikes, very big ones? What's your idea about that? First, are they on the left or on the right side? Well, this is
Left.
Left side. So, what's the most likely source of those activations?
We can look at the
We were stimulating on the left side. They are very early and large components.
You see, if I measure the latency, the amplitude of these components, in this case, and it's not one of the largest, is more than 250 microvolts. And I measured this one, which is not the largest.
Decay or a TMS?
Well, let's see. Okay.
I wanted to say just, there is a decay because this has the waveform of a decay. But before, so there is one. But before the decay, what's this? This is biphasic and sharp because, say, it ends around 7 milliseconds.
A scalp muscle artifact?
Sorry?
The scalp muscle artifact?
Precisely, yeah. Yeah.
So why you are saying so? Give it back to why you are saying so? Say it again, please.
That looks like the signal that I usually get when we stimulate muscle. So.
But from the very periphery, right? This is technically is periphery too, but we are closer to.
Much on the left side.
Okay, great. So the waveform is similar to the motor evoked potential you get from, say, muscle of the hand when you stimulate on the other side of the brain, right? The primary motor cortex, if you are in the right spot, right? Yeah, but in this case, you have the same waveform on the same side of the stimulation coil. So this is coming from a direct activation of a scalp muscle. Okay, great.
And in addition, it occurs not after 20 milliseconds after the pulse, but immediately after the pulse. That's why we say it's a direct activation of skull muscles.
Usually the latency of peripheral motor evoked potential is around 20 milliseconds, roughly a bit more, like 20, 22, 23 in a healthy subject. Okay, this is right after the pulses Silvia was highlighting. Okay, thank you, great. So let's move on. Now, the aim is to look for a response, and before that, since in this case, this decay that is coming after the motor evoked potential that is most likely due to the motor evoked potential itself, it is basically covering the response of the brain to TMS. We have to do two things. Next steps are the following. First, trying to reduce or even abolish the muscle artifact. Second, looking for a EEG response to TMS. To reduce the muscle artifact, we can start playing as described in the Silvia's paper, RTTAP 2022. We can start, and from the, actually, it is this procedure is inspired by the paper from Thomas Mutanen, who is also here. Can you shake your hand, Thomas? Okay. In this paper, Thomas was suggesting that to reduce the scalp muscle artifact, you should systematically rotate the coil, so changing the coil orientation to excite, to avoid the excitation of scalp muscle fibers. So we are going to try in this procedure.
And we were lucky we can spot immediately the presence of a muscle artifact because we masked the pulse artifact first. If we were not able to mask the pulse artifact, we would have not noticed the presence of the muscle artifact because it's very close to that one. I can show you now. And so in order to see the presence of muscle artifacts, it's important to mask the pulse artifact first. And one general suggestion, so the duration of the pulse artifact is fixed. I mean, it may depend on your amplifier settings, on your TMS machine, but once the hardware and the hardware settings are not changing, the duration of the pulse artifacts is always the same. So if you see something spiky occurring after the usual duration of the pulse artifact, you might be tempted to lengthen the window for cutting out the pulse artifact, but that's a wrong approach. You should keep that window the same, and it will be, you know, another artifact, a different artifact, something that you have to take care in a different way, not simply masking it. Okay, so here if I switch off the feature that allows me to mask the pulse artifact, I cannot see anything. Whereas if I turn on back the erasing of the pulse artifact, I can clearly see that there is an additional artifact occurring there, which is of muscle origin. I should treat it in a different way. Since we found a good spot on Gabriel's brain, we can now tell the neuronavigation software to help us in going back to the same spot exactly, because we were happy with that, or at least we wanted to start changing stimulation parameters starting from there. So we have, say, set the stimulation point such that from now on, Mario will not look at the brain anymore to search for the stimulation spot, but now Mario will move the coil relying on this virtual aiming device. So first he will move the coil such that a ball will center exactly this hole in the middle, and then Mario will adjust orientation and tilting of the coil until it will perfectly match the settings that we were applying before. Once everything will be set, the ball will become green, and so we can be sure that we are stimulating exactly in the same way. This is very important because if you run a long session, even when you ask the subject, please don't move, also the operator moves. So across trials, you may change your original spot. And if you want to repeat the same stimulation parameters over time to test the effects of a treatment, for example, so you go back 1 week later and you want to test the same EEG response, it's very important to be able to find exactly the same spot on the head. So now, just remember the muscle artifact on EEG responses to TMS. We can clear the average and okay. So Mario, you wanted first to rotate the coil and then to lower intensity maybe for avoiding peripheral reafferent signal. Okay, so now the ball is obviously red, but because Mario specifically wants to change the rotation of the coil in order possibly to get rid of the muscle artifact. Okay, when you want. There is a location control. Ah, sorry. Okay. It's possible in this case to fix the stimulation parameters and to ask the stimulator not to deliver pulses when you have the ball red. But in this case, we want to change stimulation parameters, so we have to allow for that. So now we can look at the response, but still we see a huge muscle artifact on the left side of the scalp. So that rotation wasn't enough. Let's clear the average and try to rotate a bit more. Again, a lot of muscle artifact and maybe worse than before. Again, muscle artifact. It could be that the intensity is very high. Again, muscle artifact. Yeah. So far, Mario tried to change the rotation clockwise. Now he's trying counterclockwise. And you can see that a very slight rotation significantly reduced the amplitude of the muscle artifact.
Silvia, were you here? Okay.
So before, the amplitude was more than 250. Now the amplitude is, okay, 80 microvolts. So we already have improved the situation, although muscle artifact is still present. We can try to further rotate. Do you want me to set the last stimulus? So let's set the last stimulus, which was this one, and start from there. So this is our new starting point. So this was the same as before, just clear a bit and try rotating counterclockwise again. Well, still good, a bit of muscle, but better than before. Maybe a bit less. Maybe one before the last. No, I would take this. The best was this one, right? Okay, so this was the best. Since Gabriel is still moving, we can take this spot and lower a bit stimulation intensity.
So we still have the muscle twitch in the periphery. We want to get rid of this too, okay? So now we have the minimum amplitude for the scalp muscle with this spot and with this rotation. Of course, during the hands-on, you can see it, you can try this procedure on your own, and you can play with it a bit more. Now, for the sake of time, we keep this spot with this rotation, and we reduce the intensity of stimulation in order to abolish also the peripheral twitch, okay, and to avoid the sensory reafference.
Why rotation is helpful for reducing muscle artifacts? Because if you stimulate with the induced electric field orthogonal to the nerve ending terminal, you are maximally effective on the nerve ending terminal. But if you change the orientation of the induced field, you might go parallel. So you are less effective on the nerve ending terminal than before. We lower intensity. Let's go at 45. For example, let's try. Ah? Pensavo di averlo messo. Ah, è questo qua. Ok. Now you can still see that there is a bit of muscle artifact left, very brief, very spiky. you don't see it. Okay, just wait. So Mario will deliver, as we did before, 20 pulses. Sorry, Mario, I did not put you the location control, but you were quite still on the spot. Okay, now Mario has delivered 20 pulses. So it's true, there is a residual muscle artifact, but we can start looking and see whether there is an evoked response, which means oscillations in the post-stimulus larger than in the pre-stimulus, especially near the location of the TMS target. So we were stimulating the primary motor cortex, so electrodes would be, the closest ones would be CP1, C1, CPZ, CP3, so that row. I don't know what Mario just zoomed, but I think the rows is that one. C1 and CP1, okay. Consider that each trace that you see is from -100 to +400 milliseconds around the TMS pulse, which is delivered at time 0. Okay, so, sorry, I go here. So if you look at this waveform, you see there's a spiky activity, still a residual of muscle. But then, this is pre-stimulus, which is already quite flat, already after 20 trials. From this on, it's post-stimulus. And here I can measure the latency and amplitude of this component. So latency is 28 milliseconds. Amplitude is a 6.1 microvolts. The following one is 58 milliseconds with an amplitude of about -15 microvolts. So we have a peak-to-peak amplitude of, say, about, 20 microvolts overall, so +6, -15, so above 20 microvolts peak-to-peak, in the first, say, 60 milliseconds after the pulse. So we have a response with a huge signal-to-noise ratio already after 20 trials. We may think about, in this case, reducing again intensity, stimulation intensity, because if you reduce stimulation intensity, perhaps the residual muscle artifact would go away, but we would still have a nice evoked response because this one is already very big. I would proceed in this way. If you think about other strategies, just let me know and we can comment. Of course, how can I say that this response is genuine, generated genuinely by the brain? Because, so first of all, more or less I'm stimulating here. So this is a CP3, right? So if I zoom over this channel, I see that the largest response occurs early in time. Then if I compare CP3 with the homologous channel on the other side, I see that the response is there. There are some oscillations appearing in the post-stimulus, but they are much smaller. And since I'm stimulating on the left, I'm expecting larger responses on the left side. And also, let me zoom over this set of central channels. So, just to give you a reference, this is CZ. So you see that this channel, which are close in space on the scalp, they have very different amplitude response, which is very much consistent with where Mario was stimulating. That's why I assume this is a genuine brain activation evoked by TMS pulse. Okay. So we can try already. Okay, so Mario just lowered intensity at 40. I can put location control if you wish. And then I clear the average, and let's see what happens. It's right, still a muscle, but already after 9 trials, still a large response. So let me zoom, sorry, again around, you know, the 3 channels I was showing before. So here we have 5 microvolts at 24 milliseconds and minus 12 microvolts at 50 milliseconds. So still we have 18 microvolts peak to peak as a response. So we can lower again intensity. Well, why should we lower intensity? Of course, first we can reduce muscle artifact, perfect. But then, well, what's bad in having a high signal to noise ratio? Nothing is bad, but if you stimulate at a high intensity, you are actually stimulating a large cortical area because the volume of cortex that's being activated suprathreshold is much larger. If you can reduce stimulation intensity but still get an evoked response, you are much more focal on the circuit that you want to target. So, now you see that the muscle artifact is not present anymore, but the response is still there. So with a similar, say, spatial distribution. So let me zoom over this area. This is CP3, which is still the largest channel together with CP1. You know, these two channels are the largest. So let me zoom over CP1 as I did before. You see that here we have 1.8 microvolts, 26 milliseconds, and minus 9.7, so about 10 microvolts peak to peak at 44 milliseconds of latency. Now we are much more specific on the motor cortex. And still, even if we have lowered the TMS intensity, we have a response with a very good signal to noise ratio. So you see all of this and it seems kind of magic, but behind this display there is just a masking of the pulse artifact. I can show you here, you see it's just flattened, and the time window that's flattened is between -2 and +5 milliseconds. That's it. And then behind this, there is just averaging across trials and re-referencing of each channel to the average reference. So in reality, we are recording something like this. So let me zoom over a region of interest channels. Okay. This is average across trials, but each channel is referenced to the physical reference that we put on Gabriel's head. So you see that there is something in all channels, but the waveforms are very correlated because, of course, they are all picking from the same reference electrode as a common signal. So we can get rid of this, say, similarity across channels by re-referencing everything, each channel, to the average of all channels. This small trick is very important because it allows you to appreciate where the largest response comes from which channel it comes from. And it's very important to be able to include in the computation of average reference only good channels. So imagine that for some reason there was a channel that is broken or dried out or full of line noise because it was not well prepared. If we compute average reference including that channel, it would mess up all the other channels. So in this graphic user interface, we can say we don't like this channel on top, and we decide, okay, to switch it off. So it will be hidden from the view, but also from the computation of average reference. And this is very important to be able to make these decisions fast in real time in this graphic user interface. Of course, everything, all the raw data, original ones, are recorded on the hard disk. So you will have offline to remove the pulse artifact again, to rerun average reference, filtering if you like. Here there is no high pass, low pass filtering, nothing. Data are from DC to 1 kilohertz and sampled at 5K. That's it. Of course, since we stimulated the primary motor cortex, as I told you before, the largest response is here, around CP3, CP1 channels. If we now move the stimulation coil and we go and stimulate other cortical sites, we would like to see also largest responses in different channels. They should follow our coil. And this is what we are going to try. Now we clear the average. And here Mario, on the MRI, selected a putative target for stimulation. Okay. So in this case, differently from the motor cortex, we don't have a peripheral twitch if we stimulate premotor cortex. So we don't know at which intensity we should stimulate. What we usually do is to set stimulation intensity first based on the intensity, estimated intensity of the induced E field on the cortex. So I guess that with this skull to cortex distance, about 18 millimeters, let me see if it is correct, yeah, a bit. So I think we should go at 60% of the maximal stimulator output in this case. It may vary across stimulators. So here you see in red, here, you don't see it in red. Well, here, believe me, you see about 100 volt per meter. You know, around 110, 120 volt per meter, you should be already effective on the cortex. It's 100. Let's try and see what happens. So it's not 20 volt per meter, right? We go for that. Okay. Okay. So we can see that there is a muscle artifact there. And in the post-stimulus, there are a lot of large oscillations, I would say. Sai cosa? Secondo me bisogna... Ok. Ah, perché zoom, ho capito. Ho capito. Let's just wait a bit. So we get, okay, 20 trials as we did before. So again, there is the spiky activity, but so there is still spiky activity, but it's more frontal compared to before, a little bit more frontal. And also, if I zoom over channels, I see that now, so before it was very clear that the largest response was here. Now we see even larger components over more frontal channels. Here we have FCZ, for example. Let's look at the amplitude of these components. Let's take this channel, which is cleaner from artifacts. This is 6 microvolts. This is minus 15. Again, we are very, very high, maybe in stimulation intensity. So let's go lower, and let's hope that also the muscle artifact will improve, because we want to be focal. What I told you before regarding the motor cortex, you see that in this case we are stimulating more or less here, and we see a response which is large here. But also, if I go on the back, I still see quite large responses. This means that I'm maybe stimulating an area that involves different neural circuits that spread activation all over the brain. So I'm not very focal. I'm not probing a specific circuit. Okay, so let's lower intensity. Let's go to 50 or, yeah. 20. Okay, let's set the stimulation parameters.
When Silvia says we are setting the stimulation parameters, this means that we are actually storing the coordinates of the stimulation parameters of that specific stimulus. That means orientation, intensity, tilting of the coil, and we reproduce exactly those parameters, okay?
Now we just wait again about 40 seconds. Let Mario gets to 20 pulses. Okay, 20. Now, you see that the muscle artifact is gone completely, and we can zoom over this set of channels, and you see that now the largest response, the largest response is more frontal. So let's take first this channel, which is CZ. Before, the largest set channel was CP1. Now we take CZ, for example, and here we have 2 microvolts at 21 milliseconds, minus 11 microvolts at 34 milliseconds. So the amplitude is still quite large, but muscle artifact has gone. There are several oscillations in the post-stimulus, and latency has changed a bit here over the premotor cortex as compared to the primary motor cortex. Now we have, as I told you, 22 and 33. So before, the first component was same around the 24, maybe something. But the second component was around 50, 45, 50. So it means that the waveform that we see changes with the stimulation site, which is clearly expected because we are probing different corticothalamic circuits.
I have three comments. Well, first, sorry for the quality, for the bad quality of the pictures on the screen. This is due to the beamer, but actually the recorded movie will have a perfect quality since it is not coming through the beamer first. So if you want to go then back, well, first you will have the chance to see closer tomorrow and after tomorrow during the hands-on what we are doing now, and you can again try it on your own, and then you can go back to the movie that Liam and Shiva are shooting now, to the recording that they are shooting now, to see again what we are commenting on and the images we are showing now. The second comment is about the center of mass of the response to TMS. So if you look at the whole, if you look at the whole skull picture here, the center of mass of the response was moving from here to here and was going along the changing of the location of the coil. So we first targeted the primary motor cortex, then we moved like 2-3 centimeters just toward the front, more frontally, and the response was changing, was slower for the primary motor cortex and faster for the premotor cortex, with the center of mass moving along the coil location. So center of mass here for the primary motor cortex, for the premotor cortex was more frontal. Now, to stress even more this aspect, we will target, and this I think is going to be the last session of the day, we are going to target the occipital cortex, which is very interesting for many reasons, under many aspects. For instance, Gabriel is running a study on blind people, and that's actually TMS-EEG is the only way to measure cortical excitability in these people, since the sensory pathways, Marcello was referring to that in his talk, the sensory pathways is not working, so you cannot test for the occipital cortex with the visual evoked potentials because the retina or the retinogeniculo-cortical pathways is interrupted, but you can test directly by stimulating the occipital cortex, the cortical excitability in these people. So we are going to, you wanna, just.
So, sorry, Mario, can you? So before I zoomed over CZ, but here you see that this channel has a sort of isolated artifact which is present only in this channel, is not spread everywhere, so it's not muscle artifact, and it also has a different shape compared to before. It's not biphasic, so it's monophasic and then goes back. This is a decay artifact. You know, it happens. Over time, you know, impedance gets worse. So if this was a real recording, a real session to be recorded, I would stop here, go there, correct impedance at that channel, maybe add a bit of gel, retry it, and I'm sure it will be over, this artifact. So it's very important because in real time you can correct for these tiny but significant problems when you have to clean them during the pre-processing. So it happens, it's not a problem. Okay. Now we move towards the occipital cortex. Now, let me, if I don't see the Q news, I cannot. Okay. Let me zoom a bit on the cortex. Okay. I clear the average and I turn off the location control and maybe move the camera, because in this position, poor camera is not seeing the coil anymore. Okay, great. Now stimulation is 50. Well, again here the maximum induced electric field is about 100. We can try, but yeah, we can try at the same intensity. So before we were stimulating the premotor cortex at 50% of the maximal stimulator output. And even before we were stimulating the primary motor cortex at 40 maybe, or 35. Okay, luckily we don't have muscle artifact in this case, so we can proceed with averaging. Okay, now we have averaged 22 trials. I can zoom over a set of channels. And you may see that these frontal channels are a bit flatter than before, and the ones with larger oscillations are here on the back. I can zoom over this channel, which is a PO3, and you can see that here we have a large response, peaking at 25 milliseconds, 10 microvolts amplitude, and then the second peak is sluggish, is around 74 milliseconds, minus 5.8. So, first, at the same intensity in terms of the maximal stimulator output, when stimulating the occipital cortex, we got a much larger response compared to when stimulating the primary motor cortex. So excitability of these two cortices can be different, and also scalp to cortex distance can be different. So same maximal output, but different effect because of a different distance. For example, we didn't show it today, but when you stimulate the parietal cortex, usually the skull to cortex distance is larger compared to premotor cortex. So usually when you stimulate parietal cortex, you have to increase stimulation intensity to get the same effectiveness on the cortical surface. Second change. So amplitude is one thing, and latency is another thing. So before, you remember, first peak here is 24, more or less as the one for the motor cortex. But the second peak here is 69, 70. So much later compared to both premotor but also motor cortex. It means that the EEG oscillations evoked by TMS peak at a lower frequency when you stimulate the occipital cortex and at a higher frequency when you stimulate more frontal cortices. This is again a characteristic that is specific for the stimulated circuit. And we can use it in real time to evaluate whether the EEG response is genuine or not, especially if we are dealing with a healthy subject. Of course, with a patient, you know, brain injured patient, very strange patient, you never know what's going on. But at least you always measure the amplitude of the early evoked responses close to the stimulation site. You compare this amplitude with the amplitude of later components of other channels far away from the stimulated site. And also in this case, you can appreciate the asymmetry in amplitude. So here we have a larger response, PO3, and a smaller but still present one in PO4.
Just a little comment. Yeah, if you zoom on in that channel, yes. So you have clearly a burst of oscillations in the alpha band. So based on the interpeak latencies that Silvia was reporting a moment ago, these oscillations are in the alpha band range. But first, the subject is eyes open. So we are triggering rhythms that the brain can do, especially when the subject is with his eyes closed. So with TMS, as Marcello was saying, we are, and by perturbing the brain, we can see something that the brain can do, but is not doing it in that specific moment. And second, this alpha rhythm is asymmetric, is larger, more apparent on the left side where we are stimulating rather than on the right side, while the spontaneous alpha rhythm that you can get, the Berger's rhythm that you can get when the subject is with the eyes closed, is widespread all over the parieto-occipital lobes. So we are actually forcing the brain to do what it can do, but it's not doing in that specific moment. And this is also the reason why we are using that in specific and special conditions, such as, for instance, in blind people. Actually, maybe Gabriel wants to comment on this. There are interesting results in blind people, like you can maybe report them. So when you stimulate the occipital cortex.
As you see during the demo, you saw a gradient from the occipital to the frontal cortices, meaning a faster oscillation in frontal and slower in the back. What we see in blindness, it's not a shifting, but we lose this gradient. So we have a response that it's really similar to the parietal, to the premotor cortex TPs in the occipital cortex of the blind individuals. And the interesting part is that we see this plasticity changes not only in congenital blindness, but also in late blindness, meaning that it's probably something that builds up after years and years of blindness. It's not something that is strictly tied to early age plasticity. And the thing is that something so simple as a wave shape can be really indicative in massive shift in something that's happening in the brain, be plasticity with blindness, be slowing with stroke, be slowing in disorder of consciousness. There are a lot of complex measures that you can do on that kind of signal, but the really power of the graphical user interface is that if there's a really big changes between one condition and another, or one patient and another, you can really see it online. So the cool part is that with blindness, we knew we had our results even before analyzing the data, because we can see the TPs on the screen. And when you control for any kind of artifact, muscle artifact, or other kind of artifact, if you are sure that you have a clean signal, you can infer something even online during that same experiment. So it's really helps to stay close to the data and then make also some inferences on those kind of intuition you have during an experiment.
Did you have the chance to assess people relatively acute after blindness?
No. It's something that we want to do. And for now, we will stop with our group because it's almost over. But it would be also interesting to see people that are not completely blind, that's some kind of remaining vision, to see if that remaining vision can somehow impair the plasticity. And also there's also cortical blindness with hemianopsia. There's a lot of interesting stuff in occipital cortex.
Do you have other questions, something you want to see, to ask? Please, can I?
Maybe can you show us again the whole patterns when you dezoom? So I have two questions. First one, what you see on the contralateral side, so right side, and the potential, how can you say those are evoked potential or direct cortical response, but more distant from the stimulus? Because when we do that in intraoperative monitoring, so I've done a little bit of work with Riki Matsumoto from Japan. We measure evoked response following direct cortical stimulation during surgery. And we can observe evoked, so what we call CCP, corticocortical potential. So with what we call P0 or P1, the first depolarization, positive. But we have usually a little shift in the temporal aspect, because it goes through the white matter and does the evoked potential. And if we have simultaneously a response, this is possibly the stimulus that goes through the cortex and not through the white matter. Can you assess that in this pattern, specifically left and right occipital response?
Let me try to zoom here.
Yeah.
In this case, the latency of the first peak is 23 milliseconds. If I go on the other side, let me check, it's 33 milliseconds. So visually they seem at exactly the same latency, but they are actually a bit delayed. So in this case, I think that this response, which is earlier, has been directly evoked by TMS, whereas this one comes because activation of this area might have traveled through, I don't know which pathway. So white matter or cortical-cortical, there it depends on the cortico-thalamocortical, it depends on the latency, of course. Adding information about, for example, DTI could help in interpreting the results. And of course, we cannot stay at the scalp level, but we have to go with source modeling.
So my second question is nearly the same. Can you check the very anterior frontal response? And is it possible, or did you notice in your experience that you can elicit what we call CCP, but very long-range CCP, for instance, here via the IFOF, inferior fronto-occipital fasciculus?
Yes. So here you see that in the post-stimulus there are oscillations that are larger than the pre-stimulus. So of course we may activate some circuits that then travel towards the front. And it's very common, for example, when you simulate parietal cortex, to see activations also in the frontal cortex. When you look at scalp data, you always have also to be careful about volume conduction effects. So the waveforms can be very much correlated across neighboring channels because of that effect. But of course, in this case, we are looking at frontal channels, F1, this one, for example, and we were stimulating over the occipital cortex. So in this case, I would not say it's a problem of volume conduction. I would trust this activation as something genuine, like an indirect activation of cortical sites that are far away from the original one, the first one that's been activated. And of course, connectivity plays a role in this case.
So we can say that we activated the IFOF from occipital to frontal with this pattern. You think it's related to white matter?
Yes,
okay.
In this white matter or cortico-thalamocortical connections.
Okay.
Sorry. Yeah, of course. I mean, volume conduction plays a role nonetheless, so you cannot conclude anything from scalp straight, but Marcello showed a video in the morning, coupling TMS-EEG at the source level with DTI showed a sequence of activation that were specific for the site of stimulation. So in sense, if you stimulate frontal areas, it follows a pathway that is, let's say, explained, so the variance of which is explained by the DTI tracts connecting those areas, which is different from the occipital stimulation, which follows another path, and so on and so forth. So in principle, yeah, I mean, it doesn't have probably the specificity of the CCPs recorded by bipolar, very local LFPs, but yeah, in principle, you could map it.
Yeah. On the other hand, it's a great topic. It's a great question because it remains that even we can safely assume that these are inferred, that these are long-range, genuine activation. But the question whether they are feed forward or to the extent this is feed forward, like a N1 CCP, say, or reverberant activity in the circuit, that's still open. And modeling suggests that after 100 milliseconds you start having reverberant activity feedback, but we need more data and a more direct assessment of this. One other comment is that we recently compared with EEG responses to TMS and intracranial electrical stimulation, not in the same subject, different patient, but with a common readout of EEG recordings. They're very different. They're very similar in some respect, but also very different. What we observed is that the intracranial stimulation, delivered at the Niguarda, parameters, that is, I think it's between 3 and 5 milliamps, one shock, short, I think it's sub-millisecond, no, it's 1 millisecond, whatever. But you have an initial activation, then a prolonged inhibition, at least locally, and then activity resumes, but it's a way simpler response in wakefulness as compared to TMS-EEG. And this is an important point for TMS-EEG. I think TMS-EEG is gentler on the circuit as compared to intracranial bipolar electrical stimulation.
Any other question? Okay. I think we are done with the, let's free the subject and feed him.